Welcome back, Biology 205 students. This lecture, Part 1 and Part 2, are intended to discuss, we're going to discuss microbial pathogenicity and microbial epidemiology. These are chapters 15 and 16 in your textbook, but I don't necessarily recommend that you read those chapters. You probably get all the information you need from the slideshow, but you can read those chapters if you feel you want greater detail. All right, now microbial pathogenicity, the word pathos means to suffer. It's an ancient Greek word meaning to suffer, and the word genicity or gen means the cause of. Right? So the word pathogenicity literally means things that cause you to suffer, things that are the cause of your suffering. So there are, as we mentioned in the first lecture, about 95% of the species of microbes in the world are actually either they do no harm or they're actually good for us. Whereas 5% of them give the other 95% a bad name because 5% of them are actually bad for us. 5% of the microbes are actually pathogenic, meaning that they cause us to be sick. And we're going to discuss how they do that and why they do that and how they do that, including a discussion of something called virulence factors. So virulence factors, you've probably heard, for instance, that every now and then you'll see a report on the news that a bunch of people died from E. coli poisoning by E. coli. And at this point, you also know that the human colon contains billions of E. coli, right? And so E. coli in and of themselves cannot be that bad because we have billions of them in our colon and they don't make us sick. They would make us sick if we ate them, but they don't make us sick just living in our body. But when they talk about people dying or getting sick from a case of E. coli food poisoning, what they mean is that there's a particularly bad strain of E. coli that gets inside that has contaminated the meat. I hate to tell you this, but all meat is contaminated a little bit by E. coli and other bacteria. And if you eat a little bit of E. coli, it does not harm you. But if you eat a little bit of some particularly bad strains of E. coli, which are referred to as virulent strains of E. coli, that can make you sick. That could even kill you. And we will discuss the virulent strains of E. coli in much greater detail later on. But anyway, the point is that there is more than one strain of E. coli. Some strains of E. coli are relatively harmless, Escherichia coli. Some strains of Escherichia coli are relatively harmless, while others are virulent. Virulent means more harmful or harmful in smaller numbers than the regular strains. So virulent strains of bacteria and other microbes refers to particular strains of a bacteria that are particularly bad for us. Okay, so we're going to talk about what virulence factors are, and virulence factors are the things that make one strain of bacteria of the same strain. The same species of bacteria can be good or bad depending on whether it has virulence factors or not. And the virulence factors are the things that make it particularly bad. So there are good strains, harmless strains of E. coli, and there are virulent strains. The same thing is true for every other type of bacteria. There are virulent strains of other bacteria. Okay, so we're going to discuss what is it that makes some particular strains of a microbe more virulent than others, and why some of them make us sick and how some of them make us sick. And we're also going to discuss the science of epidemiology. Epidemiology is the study of how are diseases distributed amongst populations. Epidemiology is a cross between microbial pathology and demographics. If any of you have studied demographics, you know that demographics is the study of populations and how populations are distributed amongst various places. How many people live in the city versus rural areas, what ages are all the different people who live in different places, how many of them are male versus female, how old are they, what are the age group breakdowns. Those things are referred to as demographics. And if you are discussing the demographics of illnesses, which people are affected by these illnesses, where are these illnesses found, that's referred to as epidemiology. And with the COVID-19 pandemic that we're going through at the moment, epidemiology is a very important science. We need all the epidemiologists we can get. And maybe some of you, I hope some of you may become interested in epidemiology and make it your career. The career of being an epidemiologist usually involves you work for the government and you keep track of diseases. And do you see outbreaks of disease in a certain area? And if you do, how do you contain them? How do you control them? Those are things that epidemiologists worry about. And you can get a PhD in epidemiology. You can get advanced degrees. You can do research in labs. You can work for governments. It's a good profession. Because it's a government job, we don't need too many of them. We don't. It is possible to have an abundance, a glut, an overabundance of people with qualifications to do particular jobs. So you have to be careful. It's not terribly easy to get a job as an epidemiologist, but because governments will only hire so many of them, and universities will only hire so many of them. But it is a job, and it is a good job. All right, so we're going to concentrate on microbial pathogenicity in this lecture, lecture 3, part 1. And we're going to discuss epidemiology, concentrate on epidemiology in lecture 3, part 2. Okay, so as I said, the term pathogenic comes from the Greek root word pathos, which means to suffer. Pathos is an ancient Greek word meaning suffering, and then genic means the cause of. So pathogenic means something that causes you to be sick or something that causes you to suffer. The word pathos means to suffer. The word logi means to study. So pathology is the study of things that make you sick, things that make you ill. And pathology is another profession that you can go into, or another academic discipline that you can go into. And epidemiology is the study of how diseases are distributed amongst populations. Right? So who catches the diseases? What do they all have in common? Many times a disease will spring up, will suddenly appear, and people have no idea what's causing it. They don't know whether it's caused by a virus or a bacteria or radiation poisoning or something else. And so the epidemiologists have to work sort of like detectives. They have to work sort of like detectives to ask questions like, if you look at all these people that have contracted this sickness, what do they all have in common? Are they all men? Are they all women? Are they all over the age of 80? Are they all under the age of 20? Do they all drink water from the same fountain? Do they all get their food from the same store? And if you can answer those questions, and if you can get some results, if you can get some data, that will help you to crack the puzzle. It would help you to solve the mystery. And so that's one of the things that epidemiologists do. All right, now don't get too worried, because we're going to cover the names of a lot of different types of microbes in this lecture. And this is not the first time, this is not the first time that you've heard some of these names, and it will not be the last time that you hear these names. So if you have time, start studying these names, because you will have to recognize them on the quizzes. We'll have all kinds of quizzes where we ask about these different microorganisms. That's the purpose of this course. But don't worry too much, because we're going to discuss these organisms again and again throughout the course. Right? So we're going to discuss diseases that are caused by bacteria, specific different species of bacteria, different species of yeast and fungi and protists, as well as helminths and viruses. So let's begin with the Chapter 16 material on microbial pathogenicity. All right, so first of all, let's talk a little bit about diseases and how they are defined. Okay, so first of all, we're going to talk about classification of diseases and the terminology that is used to classify diseases. And there's a wonderful organization, which is a wonderful international organization, which has helped us in that respect, which is called the World Health Organization, or the WHO, WHO. The World Health Organization is a branch of the United Nations. It's being vilified in the news media by a lot of very cynical politicians these days who are trying to whip up nationalism in their own countries by saying we don't want interference from international organizations like the United Nations or the World Health Organization. We don't want these foreign influences in our country. We'll take care of our own problems ourselves, and we can't trust these international organizations anyway. They're filled with foreigners that have different agendas than we do. So this is a very cynical play by politicians, which has been used again and again throughout history to try and increase their own support by ginning up, as they say, ginning up anger or distrust of foreign organizations, right? So, but it's all nonsense. And the World Health Organization is a wonderful organization that has helped the world to rid itself of many diseases. And the World Health Organization has created, the World Health Organization, being a branch of the United Nations, it's composed of doctors and epidemiologists and pathologists from all the countries that belong to the United Nations. So this is not just an American thing or a Canadian thing or a British thing or a French thing or anything like that. There are doctors in the World Health Organization from every country in the world, virtually. And they have agreed upon something called the International Classification of Disease, the ICD. And so we will discuss that a little bit. Then, in the second part, we're going to go back in history to the 19th century to a physician named Koch, who developed in the 19th century. The idea that diseases were caused by microorganisms was a new thing. It was a totally new idea, and so people wanted to they wanted to know, physicians and medical people wanted to know, well, how do you tell the difference between a disease that's genetic or which is based on an injury versus a disease that's caused by a microorganism? And so a physician named Koch postulated these postulates. He came up with these basically principles that says, oh, you look at the person who's sick, and if you can do this and you can do that and you get this answer and that answer, it must be a microbial disease. Right? And so that these set of principles and questions that you ask were referred to as Koch's postulates. I will probably not ask you, if I ask you about Koch's postulates, I will not ask you very many questions about it, but this is something historical that every microbiologist should know about. Every clinical microbiologist should know about the origins of how people originally in the 19th century, how they went about determining that a disease was caused by a microbe. And so we'll talk about Koch's postulates. Then we're going to discuss the stages of pathogenesis. People don't just suddenly become infected with microorganisms to make them sick. There's a very specific set of steps that microbes have to go through before they can make you sick. Number one, you can't become sick from a microbe if you've never been exposed to it, right? So the first step in microbial pathogenicity is exposure, for instance, right? And then so you're exposed, somebody coughs in your face, somebody has a cold or caused by a rhinovirus, they cough in your face, you've been exposed to the disease. That doesn't mean that you'll catch a cold, it just means that you've been exposed to it. Now, second step is if it gets inside your body, it may not be able to attach to any of the cells in your body, and therefore it may not be able to invade your body if it fails to attach, So that's another thing that could go right or could go wrong. So the second stage of pathogenesis is called attachment, for instance. So there are several stages that a microorganism has to go through in order to infect you, and we will discuss those. And then we'll discuss virulence factors. Virulence factors refers to, as I said before, the things that make a microbe particularly bad, and not all members of that same microbial species will have those virulence factors. So if they have the virulence factors, it's a virulent strain. If they don't, it's not. And then finally, once you get sick, so all of this stuff has happened, so the stages of pathogenesis have been successful, the microbe has you've been exposed to a microbe that's managed to attach itself to you and invade your tissues, you're infected and you're sick. Okay, so then you're ill, and there are several distinct stages that you go through when you are sick, and we call those periods of disease or stages of illness or periods of illness. The first one is referred to as the incubation phase, for example. And many diseases work this way, that they have an incubation phase where you do not feel sick, but you're still infectious. So if it's an infectious disease, remember that we're going to talk about the difference between a communicable disease and an infectious disease. Okay, so if it's an infectious disease, which means it's easier for other people to catch, it's easy for other people to catch it from you. you go through a period that's known as the incubation phase where you do not have any symptoms, you don't feel sick, but you are still infecting other people. You're still talking to people and spraying your saliva droplets into their face and making other people sick. And then you feel sick and you realize that you have to stop interacting with other people because you'll make them sick, and so you stay home in bed. That's referred to as the prodromal phase. Okay, so then, how bad is a disease? It depends on whether it's infectious or not and how infectious it is. And it depends also on how long is the prodromal phase. So how long are you going around infecting other people without realizing it? And then you have more symptoms, and then you go into after you're over the disease, you go into a period of recovery and a period of convalescence, and those are the periods of illness. So we'll talk about those, and you need to learn those terms as well. All right, so how do we classify disease? As I said, the World Health Organization, the WHO, is a branch of the United Nations, and they have come up with a series of definitions that are referred to as the International Classification of Disease, right? And for one of the homework assignments, I might send you off to for one of the internet research assignments, I might send you off to the World Health Organization website to look up the classification of certain diseases. I might do that, or I might not. Okay, now, so the World Health Organization is the premier international health organization, the premier international medical organization. As I said, it's a branch of the United Nations. The United Nations is funded by the members of the United Nations, and the amount of money that every country gives to the United Nations, and by extension the amount of money that is given by every country to the World Health Organization, is based on the size of the economy of that country. So, for instance, the United States has the largest economy in the world, and so they donate the most money to the United Nations. China has the second largest economy, and so China donates the second largest amount of money, and so on. Canada has a relatively small economy. We donate a lot less money than the United States or China, for instance. Britain has a larger economy, so they donate more money. So you will often hear these very cynical politicians talking about paying more money to the World Health Organization than they should. That is all a bunch of political rhetoric, and that's all a bunch of political nonsense, because everyone pays money to the United Nations based on their income, essentially, right? So you'll often hear wealthy people complaining that they have to pay too much money in taxes, when in fact the tax rates are usually proportional to income. And it's the same thing with the United Nations and the World Health Organization. Every country, the richest countries pay the most, the poorest countries pay the least, and then whoever needs it the most uses it. Its services are used in the countries that need it the most. But often, if you prevent if the World Health Organization goes in and spends a lot of money to prevent a disease outbreak in a poor country, then you have countries kind of complaining and saying, well, they spent a lot of our money on this poor country, and this poor country doesn't contribute much to the World Health Organization. This is not fair. You should spend the most money in the areas that pay you the most money. And again, I think that's a false argument. It's saying that people who pay the most in taxes because they're wealthier should receive more services than people who pay less in taxes, which that argument sounds nice it sounds logical to some people, but in fact it's not. The purpose is that everyone makes an equal sacrifice, and then the government which you elect decides where those resources are needed the most. And the World Health Organization is no different. So sometimes you have an outbreak of a very bad disease that happens in a small country, and the World Health Organization will go in and spend a lot of money trying to contain that outbreak. But in fact, if they didn't contain that outbreak, it would spread, and it would certainly spread to the larger countries and the countries with bigger economies. And so the United Nations and the World Health Organization is fulfilling a very important mission by doing that. So anyway, that's just my little statement about the World Health Organization. It's taking a lot of a rhetorical beating in the news these days, and it's undeserved. It's a scapegoat. The World Health Organization is being used as a scapegoat by very cynical politicians. Okay, now, the World Health Organization invented the International Classification of Disease. So the World Health Organization is the premier international health organization. Now, if you start looking into individual countries, the United States has, within the United States, there is a very large government organization called the Center for Disease Control. And Center for Disease Control, commonly referred to as the CDC, the United States spends billions of dollars on the CDC, and they have a lot of resources and a lot of personnel, and they gather a lot of information. And as a result of that, they are a very good source of information as well. In Canada, we have the Ministry of Health, and the Ministry of Health is divided into many different sub-organizations. And you will have already done an assignment where I sent you to the Canadian Office of Public Health to look some things up. So you'll be a little bit familiar with the Canadian Ministry of Health, and so that's a federal organization for all of Canada. And then here in BC, we have the premier disease tracking organization, an organization that does research on diseases and keeps track of how many cases of each type of disease we have in different parts of British Columbia, is referred to as the BC Centre for Disease Control. The BC Centre for Disease Control is downtown it's near Vancouver General Hospital. It's only a block or so away from General Hospital. In fact, if you ever go to City Square, the shopping center City Square, the Center for Disease Control is located right next to it. And it's a very useful organization as well for British Columbia. Okay, so as I said, the World Health Organization is a branch of the United Nations. All right, now in the International Classification of Disease, we make a distinction between infectious diseases and non-infectious diseases, right? So there are some diseases that are not caused by microorganisms. The infectious diseases are the ones that are caused by microscopic organisms. Cancer, for instance, is a non-infectious disease. So you don't, if you come into contact with somebody that has cancer, you are not in any danger of getting cancer yourself because you're in contact with somebody that has cancer. So cancer is an example of a non-infectious disease. A cold or a flu is an example of an infectious disease. All right, so if we first define non-infectious diseases, there are three main types. One type of non-infectious disease is called an inherited disease, right? So an inherited disease is, for example, a genetic disease. Example, I may ask you this on a quiz, I may ask you to give me an example of an inherited non-infectious disease, and you can tell me sickle cell anemia. So sickle cell anemia is a genetic disease, and it's a mutation of the hemoglobin molecule. You know that the hemoglobin molecule carries oxygen around in the blood. And some people have a mutation to one of the globin proteins that constitutes hemoglobin, and that is an inherited disease. Okay, now the term congenital means from birth, but it does not necessarily mean genetic. It contains the word gene, but it doesn't mean that it's genetic. It is not inherited. It could just be a problem that you've had since you were developing in your mother's womb. Right? So here's an example. A good example of a congenital disease is called the congenital rubella syndrome. Now, rubella, we discussed before, rubella is a virus that causes a disease that is called the German measles. The German measles. And if a pregnant woman contracts the German measles while she's pregnant, the baby will be born with certain deformities, a syndrome that's called congenital rubella syndrome. Right, so that is an example of an infectious disease, actually, but so that maybe that's not a good example. But you've heard of congenital heart defects. If you're born with a hole between the ventricles of your heart, for instance, that's a good example of a congenital disease that is not infectious. I apologize. I've given you on this slide. I've given you an example of under the heading of non-infectious diseases. I mentioned congenital rubella. That was very bad of me. I'm sorry. I apologize for that. That's congenital rubella syndrome is an example of an infectious disease. All right, now another term that you need to be familiar with is degenerative diseases. Degenerative disease is a disease that gets worse the older you get. You would expect that as older people are susceptible to more diseases, that's true. But generally, if you have a certain disease that causes your body to fall apart more and more and more the older you get, that's referred to as a degenerative disease. Two examples of degenerative diseases are Parkinson's disease and Alzheimer's disease, which are both diseases of the brain. Alzheimer's disease affects your memory, right? So you start out by being slightly forgetful, and then within 5 years or so, you can't even remember your children's names, or you can't even remember your spouse's names. Parkinson's is a physical brain disease that affects part of the brain responsible for movements. So you have trouble coordinating your body movements, and you have tremors and you shake a lot of the time. And that shaking, those tremors, the shaking become worse the longer you have Parkinson's disease. Right? So those are examples. Sickle cell anemia, Parkinson's, Alzheimer's, those are non-infectious diseases. Parkinson's and Alzheimer's are degenerative. All right, so a disease is something that damages the body and is usually accompanied by signs and symptoms. An infectious disease is caused by a microbe, and if you catch an infectious disease, you are infected, right? So there's a difference. So if you catch a microbial disease, you are infected, and the condition that you have is referred to as an infection. That's just a little bit of grammar. All right, now, the difference between a communicable disease and a contagious disease. All contagious diseases are communicable. A communicable disease means that it's transferred from one person to another. It can be transmitted from one person to another. So one person catches a communicable disease from another person. A contagious disease is one that is easily transmitted from one person to another. A contagious disease is easily transmitted from one person to another. Example, a cold. Now, look up here. I've listed the example of a cold versus syphilis. Syphilis is a sexually transmitted disease which you generally only catch from somebody when you have sex with them. Right? So therefore it is not easy to catch syphilis. Technically it is not easy. It is communicated from one person to another, but it's not easily caught. Right? The person who catches it has to be engaging in some high risk behaviors. They have to be doing something that's slightly dangerous, and believe it or not, having sex with somebody else is a dangerous activity in one way or another. So now imagine this analogy that I like to use. If you want to decide whether a disease is contagious or merely communicable, remember, all contagious diseases are communicable diseases, but not all communicable diseases are contagious. So syphilis is an example of a communicable disease that is not contagious, and colds are an example of a communicable disease that are contagious. Now, the way that you can tell the difference is to do a little mental exercise, and what you can do is look at the example of a cold versus syphilis. Now imagine that a person who has syphilis gets onto a crowded bus and rides that bus for 2 or 3 hours and then gets off the bus. Will that person have transmitted syphilis to anyone else on the bus? And the answer is probably not. It is a communicable disease, but it's not contagious. Right? Now, on the other hand, if somebody with a cold who's coughing and sneezing gets onto a crowded bus and rides it with the same people for 3 or 4 hours and then gets off the bus, that person will probably have contaminated and therefore sickened several people that are in a close proximity to them. So a cold is a contagious disease. And so just use that little mental question. Will a person person who has this disease, they get on a bus, they ride it for a few hours, and then they get off, will they have affected other people during that time? If the answer is no, you don't think they will, then it's a communicable disease that is not contagious. If you think the answer is yes, then it's a contagious disease. All right, so virulence refers to the degree of pathogenicity. How bad is it? How harmful is it? And obviously some microbes, even within the same species, are more virulent than others. All right, now another thing that we have to look at about contagious diseases is something that's referred to as the R0 factor. This subscript 0 is also known as a naught. Naught means 0. And so the R naught value of a contagious disease refers to how many people will a person who is already sick, how many other people will they make sick on average, right? So if you have a contagious disease that has an R naught value of 4, that means that on average, if you have one person who's sick, that person within a certain period of time will probably contaminate and infect 4 other people, right? If it has an R naught value of 3, then one sick person will probably infect 3 more people. Generally, if a contagious disease has an R naught value of less than 1, the disease is what's known. It will usually basically die out, because that means that one person is not even contaminating one other person. You have a disease that has an R naught value of 0.5, for instance, it will usually die out on its own. It will be self-limiting. Okay, so one of the things that epidemiologists want to know about a disease is what is the R naught value, right? So how many, if one person is sick, how many other people will they infect? That will tell you the R naught value. So you definitely need to know what the R naught value is. All right, now here, this table shows a bunch of viral diseases, some of which you might be familiar with, and the various R naught values that they're associated with. So for instance, COVID-19, that is causing us to be holding this class by Flex remote class, COVID-19 has an R naught value of about 2.5. Now, why did I put a question mark there? It's because we're still learning about the R naught value. But from the current data, from the information we have at the moment, it appears to have an R naught value of around 2.5 or 3. Okay, what about now, COVID-19 is a coronavirus, and we've had other coronaviruses that have caused outbreaks over the last 20 years. So we have had SARS corona outbreaks earlier in the century that had an R naught value of 3.5, and we had another variation of the coronavirus called the MERS coronavirus, which had an R naught value of 0.75. So the SARS coronavirus outbreak of about 10 years ago was quite dangerous because it had a high R naught value, but we managed to. Luckily it was managed to be contained, and so it died out eventually, but not without a lot of public policy. Okay, and then the MERS corona outbreak basically stopped on its own because it had a very low R naught value. Now, look over here. Measles is caused by a virus, and it has an incredibly high R naught value of 18. That means one person would, on average, one person would contaminate or infect 18 other people. That's very significant. And that's one of the reasons why in most places in the developed world we vaccinate people for the measles, to prevent them from catching it and therefore to prevent them from spreading it. Usually in the Western world we vaccinate children against measles when they're about 13 months of age. Generally in the Western developed world, every country vaccinates at 13 months of age. Every country vaccinates children with something called the MMR vaccine, sorry. And MMRV stands for measles, mumps, rubella, varicella. So the child normally gets 4 different vaccines in the same shot that will protect the child from measles, mumps, rubella, and varicella-zoster, because those diseases are relatively contagious. Right, so you can see measles has an R value of 18, mumps has an R naught value of 5, rubella has an R naught value of 7, and then varicella, I don't have it on the list, but it's in that neighborhood as well. All right, here's some terms that you need to be familiar with. A nosocomial infection is an infection that you acquire while you are in the hospital. Usually it's the result of being cross-contaminated by another patient. Right, so if the nurse or the care aide or the doctor comes in and fiddles around with one patient in a hospital room, and then they go to the next patient and does some procedures on them without washing their hands, they will stand a good chance of taking if patient 1 is sick, they could transfer that disease to patient 2. if it's an infectious disease. So we'll talk about this later, but that's why medical professionals in the hospital are supposed to always wear gloves when they're handling patients. And so beside every patient's bed, there's supposed to be a box of gloves. When the doctor, nurse, or care aide comes in, they're supposed to put on the gloves, for a pair of gloves from the box by that patient's bed. They manipulate that patient, and then they take those gloves off and they throw them away, and then they go to the other patient and they put a new pair of gloves on from their box. If you do that, the chances are lower that you will transfer a disease from one patient to another. But that's called a nosocomial infection. It may not be the result. It may not be transferred by the healthcare worker. It may just be that the other patient coughs on you or something. But those collectively are referred to as nosocomial infections, infections that you contracted as a result of being in the hospital. All right, now an iatrogenic infection, an iatrogenic disease or an iatrogenic infection is an infection that you get as a direct result of a medical procedure. For example, you know that sometimes people become so sick that they have to have a catheter inserted into their bladder so they can urinate. They don't have the ability to urinate on their own. So there's a little plastic tube called a catheter that you insert up the urethra into the bladder. Often, if they do that at the hospital, once in a while, they will accidentally rub that little plastic tube on the skin before they insert it into the urethra, right? And you know that the skin has Saccharomyces cerevisiae, Candida albicans, Staphylococcus aureus. We find these bacteria and yeasts on the skin easily. If that catheter touches the skin and picks up some of those bacteria or some of those yeasts, they will carry them right into the bladder, and then you get a urinary tract infection, a UTI, a urinary tract infection. So those are perfect examples of a UTI is a perfect example of an iatrogenic infection, and iatrogenic infections are all examples of diseases as well. Okay, some other terms that you need to become familiar with. A zoonosis is a disease that you catch from animals. Zoonotic diseases. A zoonosis is a disease you catch from animals, either from taking care of animals or eating animal products, drinking milk, eating animal flesh, taking care of animals, and so on. Or any diseases that any infectious diseases that you catch that way are referred to as zoonoses. Zoonosis, S-I-S, is the singular form. Zoonoses, S-E-S, is the plural form. Just a little bit of grammar. All right, now, what about fomites? A fomite is an inanimate object that a fomite is an inanimate object that carries microbial pathogen. For example, intravenous needles or a dirty knife or something like that, those are not living things, but if you get an infectious microbe on a knife and you cut yourself with it, or if you, sometimes you have drug addicts, for instance, unfortunately, share needles. If one drug addict has hepatitis, it's very common for them to share a needle with another drug addict who will then get the hepatitis, and so we call the needle a fomite in that case. All right, next, a vector is something, usually an insect or an arachnid, that carries an infection from one person to another. So a mosquito is a perfect example of that. You know, for instance, that malaria is carried from person to person by mosquitoes, right? The difference between an insect and arachnid, if you don't remember from first year, they are both members of the phylum the arthropods, the phylum Arthropoda. And within the phylum Arthropoda, we have insects and arachnids. Insects generally have 6 legs and wings. Sometimes they don't have wings, and they have a separate head, thorax, and abdomen. Right. So an insect has 6 legs and 3 body segments: a head, a thorax, and an abdomen. An arachnid has 8 legs, and it has the head fused to the thorax. So a good example of an arachnid is a spider, but also a tick is a very common arachnid vector that transfers diseases from one person to another. Okay, a reservoir, in particular a non-human reservoir, is a non-human source of the disease. In the context of vectors, vectors not only transmit diseases from person to person, but they transfer diseases from animal reservoirs to humans. Example, you know that malaria can be caught by monkeys as well as humans. Right? So even if you cure humans of if you cure all the humans of malaria, people will still be catching malaria from mosquitoes, and the mosquitoes got it from the monkeys. Right? So even if you cure all of the humans of malaria, there is still a non-human reservoir for malaria, in this case monkeys. Right? So there are many non-human reservoirs. Now, what is the difference between a primary pathogen and an opportunistic pathogen? Okay, here's a very simple definition. A primary pathogen is a microbe that makes you sick or is harmful even when you're healthy. Example, Bacillus anthracis. Bacillus anthracis is a very bad microbe that will potentially kill you even if you're in the peak of health. An opportunistic pathogen is a microbe that will harm you, but only if your body is in some kind of a weakened state or a damaged state. So for example, Candida albicans, we learned that Candida albicans is on the skin surface. It's on our skin. There are billions of Candida albicans cells all over the surface of our skin. No matter how often we wash our hands or how often we take a shower and how hard we scrub, we can't possibly get rid of all the Candida albicans. Now, as long as it's on the outside of the skin, it's not harmful. But if you cut yourself and it gets under the skin, if it manages to get into the bloodstream, it can do a lot of damage once it's inside. So that's an example of an opportunistic pathogen. It's harmless as long as you're able to keep it outside the body. If it gets into your bloodstream or into your lymphatic system, it can cause damage. Clostridium difficile is an example of an opportunistic pathogen because if you eat Clostridium difficile is a bacteria that lives in soil, and it often gets on your food if the food is contaminated with a little bit of dirt for whatever reason. Let's say you're eating raw vegetables or something like that come out of a garden. There might be a little bit of dirt contamination. If you eat a little bit of Clostridium difficile, it can't colonize your digestive system. Like, for instance, we have many bacteria in our intestines. Clostridium difficile would love to live in our intestine, but when you eat a little bit of it, it gets down there and it sees that all the living spaces are full. Right? That's called competitive exclusion. Competitive exclusion. So the bacteria Clostridium difficile can't take hold, it can't get a foothold in our intestine because there are a lot of good bacteria that are already living there. But if you go into the hospital, you have some kind of a bacterial blood infection or something like that, and if they give you an antibiotic to kill the blood infection, it will also have the effect of killing the good bacteria that are in your intestine. And as long as those good bacteria in your intestine are gone, that gives the Clostridium difficile a chance to go in there and take over. Right? So that's considered to be an opportunistic pathogen as well, or an opportunistic infection. Right? Now, I should give you one caveat, one note, which is that all microbes, any microbe is harmful in large enough numbers. So what we're talking about here is microbes being harmful in smaller numbers, So we have trillions of E. coli in our colon, in the human colon, and they're okay. But if you eat a little bit of E. coli, it's generally not bad for you. If you eat a little bit of a virulent strain of E. coli, it is. But on the other hand, if you take the harmless strain of E. coli, and if you ate a kilogram of that bacteria, that would definitely kill you because it would cause a septic infection. So basically, any microbe is bad for you if you're exposed to huge amounts of it. But if you're just exposed to little amounts of it, generally they're not. Most of them are not. All right, now what is the difference between a primary infection versus a secondary infection? You need to know this as well. A secondary infection is an infection that you get as a direct result of having a primary infection or some other infection. for example, if you get a viral lung infection, that means you have an infection of the lungs by a virus. That causes your lungs to produce a lot of mucus. It causes your bronchial tubes to produce a lot of mucus. And so let's say you have influenza, right? You have the flu you have the flu, influenza caused by a virus. It causes your lungs to become inflamed, which causes your bronchial tubes to produce a lot of mucus. There's some bacteria that like to live in mucus, and so when your bronchial tubes are producing so much mucus, they might get infected with Streptococcus pneumonia. And so then you say that you have a bacterial pneumonia infection secondary to the flu. So you get a bacterial pneumonia infection secondary to the flu. So you wouldn't have had that Streptococcus pneumonia infection if you hadn't had the original infection from the influenza virus to begin with. Okay, so resident microbiota, people who study microbiology realize that they study what kinds of bacteria and viruses, what kind of bacteria and protists and yeasts mainly live in the human body, in, on, and around the human body normally. And so we refer to all of those microbes that are normally found in the human body as resident microbiota. Resident microbiota is the modern term. There's an old-fashioned term, so if you run into older microbiologists or older doctors and whatnot, they may refer to it as the normal flora of the human body. And that's an older term because the word flora means plant life, plants. And 50, 60 years ago, they used to classify bacteria and fungi as plants because it was common to classify bacteria and fungi as plants, so you would call them flora. We don't call them that anymore, right? So that's an old-fashioned term, but you might hear it occasionally. Okay, so what are the resident microbiota of the human body? They are the bacteria, fungi, and yeasts that live on the human body normally, and they're normally they are harmless, but any one of them could easily become an opportunistic pathogen if something happens to your body to weaken the state of your body. So let's look at some of the normal resident microbiota of the body, and when we go through these lists of microbes, bear in mind that any one of them could cause an opportunistic infection under the right circumstances. All right, what about the normal human microbiome, or the resident microbiome, or the skin microbiome in this case? So on the skin, you would normally find Staphylococcus epidermidis, right? The word epidermis means the outer layer of skin. So Staphylococcus epidermidis is part of the normal microbiome of the skin. You also find Staphylococcus aureus, Candida albicans, Saccharomyces cerevisiae. You know what Saccharomyces cerevisiae is because it's used in the food industry. Well, it also happens to be everywhere beyond food. You put Saccharomyces cerevisiae into grape juice to make wine. You put it into other things to make beer and other alcoholic beverages. But it's a naturally occurring yeast that you find everywhere. It's ubiquitous. You find it everywhere. Staph aureus and Candida albicans, you already know that Candida albicans is what causes vaginal yeast infections. Staph aureus can cause staph infections, which can be bad. Right now, Propionibacterium acnes causes acne. It's one of the causes of acne. So this is a bacteria that likes to live on oil. Oil is secreted from sebaceous glands in the skin. And so occasionally those bacteria, if there's a plug or a clog of some sort, those bacteria will live inside it and form a pustule. A pustule is a little tiny collection of pus where macrophages and things are coming in and trying to kill the bacteria. Okay, so these are examples of the normal human skin microbiome. All right, what about including the eyes? The eyes are considered to be part of the whites of your eyes. The white part of your eyes are referred to as the conjunctiva, And so there are some infections that infect the conjunctiva of the eyes, right? And so Staphylococcus epidermidis and Staph aureus are two common members of the resident microbiome of the conjunctiva. And they could, if they get out of control, normally they're kept in check by the tears because the tears are constantly washing them away. But occasionally, if they, for some reason, they get a foothold in your eye, you could get conjunctivitis. Remember, our definition is that if you ever have inflammation of a body part, you just take the name of the body part and you add itis to the end. So conjunctivitis is where you have an inflammation of the whites of your eyes that may be caused by a bacterial infection. In the nose, mouth, and throat, you'll have any of these things. Again, Staphylococcus epidermidis as well as Staph aureus, Lactobacillus acidophilus, which is also found in the vagina and also in the intestine. Streptococcus mutans is found in the mouth and is often caused tooth decay is sometimes caused by Streptococcus mutans. That's one of the ones that we learned from the first lecture. And then there's some other things there. Now I wanted to draw your now look down here. I wanted to draw your attention to this way of writing something. See, we have Candida Spp. The term Spp is the way you write species, right? So it means that there are a number of different species within the genus Candida, right? So we have the genus Candida, and one of the species within the genus Candida is Candida albicans. So albicans is the species, Candida is the genus. If you're talking about several different species within the same genus, you can abbreviate that by just saying Candida Spp. So Candida Spp indicates that there are several different species of Candida yeast in the nose, mouth, and throat. In the gut, in the gastrointestinal tract, you will find lots of different lactobacillus species, including Lactobacillus acidophilus. You will find lots and lots of Escherichia coli, E. coli, particularly in the colon, which is also known as the large intestine. So as the food is getting ready to go out, it's almost ready to go out, it's in the sigmoidal colon or the descending colon. That area is filled with E. coli, trillions of E. coli. There's another genus of bacteria called the Enterobacteria, genus Enterobacteria, several species of that in there. Klebsiella, including Klebsiella pneumonia occasionally, but that's a virulent strain, several different members of the Candida genus, and so on. Okay, now let's talk about, so those are examples of things that you would find in the body normally. So you should recognize some of those things if I ask you multiple choice questions about those. Now let's move on and start talking about sicknesses. Okay, let's start talking about sicknesses now. Okay, so signs and symptoms and syndromes. All right, signs are an objective measurement of illness, things that you can measure. So temperature is a great example of a sign. Red spots, you know, when you get measles or something like that, that too is a sign that you're infected with something. That's a sign of an illness. So any objective measurement of an illness that you can see and measure is called a sign. Anything that you can't measure as easily, it's very difficult to measure because it's a subjective thing, is referred to as a symptom. Right? So nausea is a symptom. It's very difficult to measure how nauseous a person is. Pain is a symptom. It's very difficult to quantify how much pain someone is in because, as you know, different people have different tolerance to pain. Just as different people have different tolerance to nausea. So for instance, somebody like me, it wouldn't take much, you know, one of the ways that people try to measure things subjectively, you've probably seen this, they use something called a Likert scale. A Likert scale is where you say, rate this on a scale of 1 to 5, right? So you say, how nauseous do you feel on a scale of 1 to 5? Well, that is a very subjective measurement, because if you take somebody like me, who leads a very inactive lifestyle, I might, if I feel a little bit nauseous, I might rate that as a 5. On the other hand, what about a sailor? You have a sailor who's on ships all the time. They probably, after a career 20 years sailing around on ships and being constantly seasick, they may have a much higher tolerance to nausea. And so what I might rate as a 5 on the nausea scale, the sailor might rate as a 1. Right? So it's very difficult to quantify symptoms. And so symptoms are subjective things that are very difficult to quantify. All right, what is a syndrome? A syndrome is a collection of signs and symptoms that are characteristic of a specific disease. And in some cases, you may not even know what the disease is, what's causing it, but that's called a syndrome. So, for example, you may have heard of the AIDS syndrome. AIDS is caused by a virus called the human immunodeficiency virus, HIV. But in the early 80s, when AIDS first started to appear, people did not know what was causing it. They just knew that certain people were coming down with a syndrome, and the syndrome was composed of anemia and being susceptible to a bunch of other diseases that are indicative or characteristic of having a compromised immune system. So they call that the AIDS syndrome, which is AIDS syndrome, which is an acronym that means acquired immune deficiency syndrome, and it was characterized by having a deficient or a damaged or a minimalized immune system. Now we know that the reason for that is because the human immunodeficiency virus invades and damages T helper cells. T helper cells are one of the main white blood cells that coordinates the immune system, and so that's how the immune system got damaged. Okay, so syndromes is just the collection of signs and symptoms that you assign to a certain disease, to a specific disease. All right, now the term asymptomatic is synonymous with the term subclinical. Synonymous means it means the same thing. So asymptomatic and subclinical means that you have a disease, but you don't have any symptoms. You may develop symptoms, right? That's why I say yet, but you don't have any symptoms right now, or you may have only minor symptoms. So that's called a subclinical infection or an asymptomatic infection. Those are two words you need to know. Now you need to know what the word etiology means. The word etiology means what is the mechanism by which you get sick? What's causing you to get sick? If you end up having blood poisoning as a result of hepatitis C damaging your liver, so your liver is no longer able to filter things, bilirubin out of the blood or something like that, that would be the etiology of that particular illness. That's how it's caused. Now, if you don't know the mechanism by which somebody gets sick, if you don't know what's causing the illness, then you say it's idiopathic. Idiopathic means you don't know the reason. So if somebody has a high temperature, you say they have idiopathic fever. That means that they have a high temperature, but you don't know the reason. Right, so idiopathic, you can memorize that word easily if you think of the word idiot. You say to your doctor, you don't know what's causing me to have the fever, you're an idiot. Okay, idiopathic means that means a symptom or a sign of unknown cause, of unknown etiology. All right, some more medical terminology. The term inflammation means that a certain body part becomes inflamed. The characteristics of inflammation are pain, a feeling of heat, and redness and swelling. So if you have a body part that turns red, swells up, it hurts, and it feels hot, we refer to that as inflammation. So something is inflamed. Now, if there's a particular part of the body that's inflamed, we describe that condition by taking the name of the body part and adding itis to the end. Itis. Right? So, for example, the membranes that surround your brain are called the meninges, and so if those membranes become inflamed, we call it meningitis. If you have bronchial tubes carry oxygen or carry air to the lungs. If the bronchial tubes become infected, we call that bronchitis. If your appendix is inflamed, we call that an appendicitis or appendicitis as it's pronounced. Right? So that's how you describe a body part inflammation. Okay, now the word lysis refers to tearing apart or breaking apart. For example, there's some microbes, there are a lot of viruses that cause blood cells to break apart, which cause bleeding, and we refer to that as hemolysis. And later on in the course, we're going to learn about a whole lot of bacteria that are classified as hemolytic bacteria. Hemolytic bacteria are bacteria that cause blood cells to burst or to be lysed, right? So, in fact, we're going to learn that there are three forms of hemolysis. There is one called alpha hemolysis, one called beta hemolysis, and one called gamma hemolysis. Beta hemolysis refers to complete destruction of red blood cells. Alpha hemolysis refers to partial destruction of red blood cells, and gamma hemolysis refers to no destruction at all. Why do we care about those words? Because bacteria one of the ways that we classify bacteria is by their hemolysis. Right? So there are a whole bunch of bacteria that we classify as being beta hemolytic. If you put them in the presence of some blood cells, they completely destroy the blood cells. And so we call those beta hemolytic bacteria. And others will only partially destroy the blood cells, and we call them alpha hemolytic bacteria. And some of them don't destroy the blood cells at all, so we call them gamma hemolytic bacteria. So just as, there are many other ways that we can classify bacteria. We classify bacteria as being gram-positive or gram-negative. That's one way we classify them. Another way we classify them is whether they are alpha, beta, or gamma hemolytic. So we'll be discussing that. And that's important. You do need to know that for the course. You do need to know the difference between alpha, beta, and gamma hemolytic bacteria, and we're going to discuss that some more later. Okay, the suffix oma means cancerous, right? So a melanoma refers to a cancerous melanocyte. That's one of the cells that gives you the brown pigment in your skin, creates pigment in the skin. If one of those cells becomes transformed and cancerous, you call it a melanoma. If you have a white blood cell, which is referred to as a lymphocyte, if a white blood cell becomes cancerous and starts dividing unstoppably, uncontrolled cell division, we call that a lymphoma. Okay, so there's another medical term for you. All right, algia refers to pain. Myalgia, for instance, meaning sore muscles. The word hepato refers to the liver. The word cyto means cells. The word pyogenic means something that causes pus to form. And so there are a whole bunch of bacteria that are classified as pyogenic bacteria. Now, I've mentioned before that pus is actually, that if you get an infected cut, you get this sticky, smelly stuff forming on the cut, and we call that pus, P-U-S. I misspelled it with two S's there. With two S's, that sounds like puss, like a cat. My wife and I have a cat that we just named Puss because we were unimaginative. Anyway, so pus is actually made of white blood cells, particularly phagocytes, that are swarming to the area of the infection and trying to kill a bacteria by eating it. And so there are some one of the functions. There's part of the immune system is called the innate immune response. That means that there are a whole bunch of white blood cells, particularly phagocytes, that are pre-programmed to attack certain types of bacteria. They don't have to learn how to do it. And those bacteria we classify as pyogenic bacteria because they provoke the formation of pus immediately. Remember the word innate immune system. The word innate means that you're born knowing how to do something. You're born with that quality. So there are some white blood cells that have to learn what a bacteria looks like, what kind of proteins it has on its surface before they can attack it. And there are other white blood cells that simply attack it because they already know. They were born knowing to attack Staphylococcus aureus, for instance. So Staphylococcus aureus is classified as an example of a pyogenic bacteria. That's another way we classify bacteria, whether they generate pus or not. Okay, you probably know many of these terms. The difference between an acute infection and a chronic infection. The word acute means temporary, usually means intense but temporary. So a cold, for instance, is an acute illness that's caused by a virus called a rhinovirus. A chronic infection is a long-term infection that you have for years or decades or even for your entire life. Many of the protists that you get infected with, you can never really get rid of. Many people are infected with malaria and they're never able to get it, so they end up taking anti-malaria drugs for their whole life. Many people get sleeping sickness because they've been infected by trypanosomes and they're never able to get rid of it. We call those chronic infections. All right, now two other terms. I want to go down to the bottom here first. A persistent infection is an infection. It's a type of a chronic infection. It's where the microbe stays in the body for a long time and causes relatively minor symptoms. So if you've got a minor cough that you have for your whole life, that would be an example of and if it's caused by a microbe, that's referred to as a persistent infection. So a persistent infection is something that's caused by a microbe that stays in your body for a long time and usually causes minor symptoms. It's an interesting thing about infectious microbes, which is that microbes that cause acute infections, either your immune system destroys them or they kill you, but either way, they don't last for very long. Whereas a lot of chronic infections, your immune system is unable to destroy them, but there are other mechanisms in your body that kind of keep the symptoms down to a dull roar, keep the symptoms to a minimum, and those are persistent infections. All right, now a persistent infection is not the same as a latent infection. The word latent means to become invisible or to disappear. Right? So a latent infection is where a microbe enters your body and it causes a fuss for a while. It causes an acute infection for a short period of time. It causes a fuss, and then it suddenly goes silent, and it doesn't make any more noise. It doesn't cause any more symptoms. It doesn't cause a fuss, and you wouldn't even know it's there. And it may stay there in this silent or latent form for perhaps decades. And then suddenly, for whatever reason, it decides to reappear and start making a fuss again. Right? So the perfect example of that is Varicella-Zoster virus. We discussed that during lecture 1. And Varicella-Zoster virus you catch it generally when you're a child, where you have chicken pox, as they're called. You have these red spots on your body, and you have fever and some other symptoms. And then it disappears, but it hasn't gone away. It's simply gone into hiding. It's simply become silent. It's gone underground, if you will. In fact, what it's doing is it's hiding inside one of the peripheral nerves that comes out of your spinal cord. And then it may, for reasons we don't quite understand, it may become reactivated when you're middle-aged and cause a disease called shingles. So we don't really know what causes the Varicella-Zoster virus to become reactivated in some people but not others. But if it does reactivate, it causes a disease called shingles, which is characterized by blisters on the body. Okay, so we'll talk about Varicella-Zoster later. All right, some other terms that we need to know about. A local infection is an infection that only affects a small amount of the body. Example, a blister. That would be an example of a local infection. A focal infection is where the infection is strongest at a certain locus, but the rest of the body is infected as well, just not as much. Right? So the rest of the body is infected to a lesser degree. And then finally, a systemic infection is where the whole body is infected, and that's an example. If it gets into the blood and it travels for the whole body, that's an example of a systemic infection. Okay, here's some other things you could learn. Okay, so bacteremia refers to bacteria in the blood, so that would be an example of a systemic infection. Septicemia means that you have bacteria in your blood that are actually alive and are actively reproducing and dividing in the blood. So not only do you have bacteria in your blood, but they are increasing in numbers. Okay, now something that's related to these two terms, bacteremia and septicemia. Septicemia is also sometimes referred to as septic poisoning or septicemia, is that we talked earlier on, and we'll talk about it again. We'll talk about the fact that gram-negative bacteria have something called endotoxins. Endotoxin is a toxic protein or a toxic carbohydrate that is it's an integral part of the bacteria plasma membrane. Right? So this poison, this toxin that the bacteria is making, is attached to the bacterial plasma membrane. It's not secreted. It's not sent out into the environment. It is attached to the bacterial plasma membrane. The only way that it will get loose is if you kill the bacteria and the bacteria disintegrates and releases all of these components into your blood. So that's a very clever thing. That's a very clever way of a bacteria infecting you and saying, I'm in here now, and if you dare to kill me, I'll certainly kill you. Right? So if you kill me, I'll do the same thing to you. So if you kill all of these gram-negative bacteria cause this phenomenon. If you have gram-negative bacteria in your blood and you have lots of them, and if you kill them all at once, the toxin will be released and cause you to go into anaphylactic shock, which is also known as septic shock. Septic shock, right? So anaphylaxis means that your blood capillaries suddenly become leaky. They suddenly become permeable, and all of the blood serum leaks out into the soft tissues of your body that causes a sudden rapid drop in blood pressure, which could kill you. If you have a sudden rapid drop in blood pressure, that could kill you because it becomes harder for the blood to transport oxygen around if it's under low pressure. So you could have a heart attack because your heart is not getting enough oxygen due to the low blood pressure. You will certainly faint because your brain's not getting enough oxygen. So if that happens, that's referred to as anaphylaxis or anaphylactic shock. And if we're talking about anaphylactic shock that's caused by bacteria, gram-negative bacteria disintegrating in your blood, that's referred to as septic shock. Okay, so recall that any microbes can be bad in large enough numbers, but if you have microbes if you have a particular strain of a specific species of bacteria or other microbe that's particularly bad in small numbers, we refer to that as a virulent strain. We say that it has virulence. Okay, so if you have a virulent strain of a bacteria or microbe, it means that very few of them can make you sick. Only a few of them will make you sick. Okay, now how many of them will make you sick? Generally, we can't. That's not something we can predict or calculate. It's determined empirically. The term determining something empirically refers to determining something experimentally. Right? So determining something experimentally. Now I want to introduce you to two values that pathologists or microbial pathologists use. One of them is called the ID50 and one of them is called the LD50. All right, the ID50 is, let's say that you're doing an experiment, so we're determining this empirically. So we're doing an experiment with guinea pigs, right? Now you've probably heard metaphorically, you've heard the use of the word guinea pig metaphorically. This is where where you're using, it means you're experimenting with a person, for instance. But in the old days, when they used to do these laboratory tests, they would literally use guinea pigs. They would use these little rodents called guinea pigs. These days, it's more common to use rats, but let's say that we're using guinea pigs. And so you take 100 guinea pigs, and then you give them you take 1,000 different guinea pigs. You take 1,000 guinea pigs and you divide them into groups of 100. And then those groups of 100, you give different numbers of the bacteria to each one of those groups of 100. And how many bacteria cells, how many microbes do you need to give them in order for half of them to become infected? How many cells of that microbe do you have to give to 100 guinea pigs before 50 of them will become infected, right? So that is referred to as the infectious dose 50. 50 stands for 50%, and then ID stands for infectious dose, infective dose, right? So generally you would do that. You would set up this experiment where you take a bunch of rats or guinea pigs and you divide them into groups, and you give each group a specific number of microbial cells, and then you see how many cells you have to give them before 50% of the test animals become infected. And once you've determined that, you have determined the ID50. So you can imagine that if a microbe is particularly virulent, the ID50 will be low. If a microbe is not very virulent, the ID50 will be very high. Okay? Now, that refers to infectious, right? So some microbes are more infectious than others, and so we say that they are more virulent, but they might be harmless. So they may be more virulent, but that in and of itself doesn't mean anything. There are a lot of viruses, for instance, that have a very low ID50, which means that the contagious viruses have a very low ID50. Means that it's easy to catch them because you don't need to be exposed to very many of the viral particles in order to get sick, or in order to contract the disease. Okay, that's fine. But what about particularly bad microbes that will actually kill you? Well, then we can conduct a similar experiment, except this time we're looking to see how many cells do we have to infect these rats or these guinea pigs with in order to kill half of them. And we call that the LD50 for lethal dose 50%. Right? So LD50 and ID50 are two measures of virulence that are determined empirically by experiment. Here's an example, right? So we say how many, in this case, cells are virions. Virions are virus particles, and cells could be bacteria or anything else. Okay, so how many did we have to infect those animals with in order to kill 50% of them, right? So here's the mortality rate in the group. It's no surprise that if like I said, any bacteria is harmful in large enough numbers, so it shouldn't be any surprise that if you give them enough, you'll kill all of them. But how many cells or how many virus particles do you have to infect them with before 50% of them will die? That will give you the LD50 number. And if you can take various microbes and you compare the LD50 values and you compare the ID50 values, and that will tell you how virulent that particular strain of that particular microbe is. So how many do you have to inject generally to kill half of the test animals? Let's look at a couple of examples here. Okay, I'm first going to show you an example of the ID50 for different types of E. coli that cause diarrhea. So some strains of E. coli are not that bad, and some of them are virulent. Okay, so the average person has about 100 trillion E. coli in their gut, right? So that's quite a few. And so the regular strains of E. coli are not virulent even with numbers like 100 trillion. Okay, there's a strain of E. coli, a virulent strain called enteropathic E. coli. Enteropathic E. coli, which is abbreviated EPEC. Right? If you eat, or somehow if 10 million of those get into your gut, into your intestine, you will get sick. You'll get diarrhea, you'll get sickness, including diarrhea. So obviously, EPEC, enteropathic E. coli, is more virulent than regular E. coli. There's another strain called enteroinvasive E. coli, which has some virulence factors that allow it to get into the cells of the intestine quite easily. That's why it's called enteroinvasive E. coli. And you only have to eat or swallow 5,000 of those to get very sick. And then finally, the worst one of them all is referred to as enterohemorrhagic E. coli, abbreviated EHEC. You only have to eat 10 of those in order to get violent bloody diarrhea. Hemorrhage means to lose blood, right? So enterohemorrhagic E. coli causes diarrhea that's filled with blood because that particular virulent strain of E. coli is actually lytic. It's a lytic, causes a lytic infection that causes the intestines, the cells of the intestine to break down and bleed. Right, so whenever you hear these news reports of so many people dying of an E. coli contaminated meat, they're usually talking about enterohemorrhagic E. coli. They're not talking about regular E. coli. Okay, so that's an example. We're comparing the amount of different strains of virulent E. coli that it takes to become infected and also to become sick. All right, now let's look at the ED50 for the infective dose for Bacillus anthracis. So Bacillus anthracis, of course, it's an endospore forming bacteria. Endospores are little tiny seeds that it makes, little tiny haploid seeds. And if you have now, how does the bacteria? So we're talking about the spores, not the bacterial cells themselves. All right, so how do these spores get into your body? There's several different routes that you can get into. I want to draw your attention to this word. You definitely need to know this word. The word parenteral means to enter your body through a cut or a break in your skin. That's called the parenteral route of infection. Respiratory, you can guess, it means to inhale the endospores. And gastrointestinal in this case means you eat it. You eat them. Okay? So it only takes 10 endospores entering through the parenteral route to give you an infection of Bacillus anthracis. It takes 10,000, you have to inhale 10,000 of them, endospores, in order to get infected through the respiratory route. And you have to eat 1,000,000 of them before you'll become infected using the gastrointestinal route, or the oral fecal route as it's sometimes called. So that's just an example. Here we're not talking, in the previous example we were talking about different strains of E. coli. Some of them were more virulent than others. So enterohemorrhagic E. coli is much more virulent than regular E. coli, and so the ID 50 numbers are smaller. Now we're talking about exactly the same bacteria. We're not talking about different strains. We're talking about all the same strain of Bacillus anthracis, but in this case we're asking, how many spores do you have to get into your body through various routes of entry to make you sick? Okay, so that's another use of the ID50 values, right? So if Bacillus anthracis enters through the parenteral route, it only takes a few cells. If it enters through the respiratory route, it takes more. If it enters through the oral fecal route, it takes quite a bit more, right? So that's an example of using the ID50 value in a different context. Okay, as promised, this is not my most exciting part of the lecture, but we do have to learn this. We're going to learn about Koch's postulates. Okay, so Robert Koch was a German physician, and in 1884, in the 19th century, in the late 19th century, the idea that certain illnesses were caused by microbes, by microscopic organisms, was a relatively new idea. And people wanted to know, how can you tell whether an illness is caused by an infectious microbe versus something else? And so Koch came up with what are known as Koch's postulates. Now, this is a set of 4 different rhetorical questions, or 4 different things that you have to do in order to conclude that something is caused by an infectious microbe. Okay, now Koch's postulates are not really true anymore, and the reason for that, these are the original things, we don't really accept them anymore, but they were originally accepted because we didn't know any better. But let's talk about them. Let's first learn them, and then I'll ask you what you think is wrong with them. What are the shortcomings of Koch's postulates? Before we can ask that question, before we can ask what are the shortcomings of Koch's postulates, we have to ask, I need to make sure that you know the difference between a necessary and sufficient condition versus a necessary but insufficient condition. Okay, let me draw your attention to the idea that you can sometimes learn what's causing something to happen by carefully dissecting different parts of something and then seeing which things you have to delete or get rid of before it stops happening. Right? So those of you that were in my Biology 200 class know all about this. It's called an ablation study. And what it is you want to figure out how something works. You want to figure out, you have some unknown organism or some unknown machine, for instance. If you want to figure out what every part of that machine is responsible for, you can first look at what that machine does, and then you can start taking out the parts one by one and seeing what happens when those parts are not there. So for example, if you saw an automobile for the first time and you know that an automobile moves forward, and then you want to see which parts of that automobile are responsible for moving it forward, you can start taking the parts out one by one. Okay, so if you take the windshield wipers off of a car, it'll still go, and so you say, okay, the windshield wipers are not involved in making the car go forward. You can remove the heater and the car still goes forward, and then you say that's fine, that's not responsible for that. If you take the battery out of the car will not go. But you and I know that that's not because the battery is what's driving the car forward. It's because the battery is an important part of the engine, and it's actually the engine that drives the car forward. Right? So you would say that having a battery, a car having a battery, is a necessary but insufficient condition for the car to move forward. If you just have the battery without the engine, the car is not going to go forward. If you have the battery and the engine, it will. Right? So having a complete engine with a battery is a necessary and sufficient condition for the car to go forward. right? So there's a difference between a necessary and insufficient condition and a necessary and sufficient condition. So some of the reasons why we think Koch's postulates are obsolete have to do with the fact that necessary and insufficient versus necessary and sufficient conditions. So we'll talk about those. All right, here are Koch's 4 postulates. Number 1, the suspected pathogen must be found in every case of the disease and not found in healthy individuals. Okay, so you have a bunch of people that have a disease. You take a sample of their blood, for instance, or their sputum. Sputum is you spit into a cup and then you look at it under the microscope. You stain the sample and look at it under the microscope. So you look at their blood or their urine or their sputum, and you ask, can you see anything in there? Okay, so you identify a bacteria based on morphology. Probably in Koch's day, the only way they had to identify bacteria was based on what they looked like. So you take a bunch of sick people, they all have the same disease, you take a blood sample, you look at their blood and you see the same bacteria or microbe or something in their blood. You always find that in the blood of the sick people, and you never find it in people who are not sick with that illness. That was his first postulate, and it sounds pretty good, right? That sounds like a perfectly good thing. Okay, number 2, the suspected pathogen can be isolated and grown in a pure culture. So you take that person's blood and you streak it on a Petri plate, and then you get an isolated colony on the Petri plate, and then you use that to inoculate a bottle full of sugary water, and then you can actually grow that microorganism in a bottle in a pure culture without any other things in it, right? And you can do that with most bacteria. You can do that with many protists as well, right? So in the days of Koch in the late 19th century, with our limited knowledge of microbes in those days, that was probably a good postulate as well. Okay, the third postulate is, okay, if you take this thing out of the person's blood, you isolate it, you grow it in a pure culture all by itself, and then if you take it and you infect some test animals, like rats for instance, and they develop the same disease that you saw in step 1, in postulate 1, that's a pretty good proof that that microbe that you saw and that microbe that you grew in a bottle was what was causing the disease in the first place. And then finally, you take those new animals, guinea pigs or rats or something, you take those new animals that you newly infected with this microbe, and you look in their blood, and lo and behold, you see the same microbe in their blood. Then if you satisfy those 4 conditions, then that is the thing. Then that is definitely a microbial disease. It's a disease that's caused by microbes, and you have successfully isolated the microbe that is causing it. And by 19th century standards, that was a wonderful breakthrough in this field of infectious diseases. Okay, so condition number 1, you always see this microbe in the blood of diseased animals or humans, but you do not see it in the blood of healthy animals, right? All right, so you take the blood of those diseased animals, and then you streak the blood out on a plate, and you get an isolated colony, and you grow it up in a flask, in a bottle, and then you can identify it as a pure culture, and then you go and you inject it into a healthy animal, and the healthy animals get sick, and then you look at the sick animals, the animals that you have just made sick, and then you look to see if the same microbe is there, and if it is, you've satisfied all 4 of Koch's postulates, and you've determined not only that this is a disease caused by an infectious microbe, but that you have identified the microbe that's causing it. That's the important part. Okay, so do we have any problems with these by modern standards? Well, yes. Okay, so first of all, by modern standards, now that we know a lot more about different microbes and things, we know that okay, let's look at step 1, for instance. Okay, so first of all, you have to be able to find the pathogen in the diseased individuals, right? You have to be able to see it. Okay, so that involves a problem with sensitivity. So then if a microbe is very virulent, that means that only a few cells will make you sick. There's a very good possibility that if you look in their blood, or if you look in their blood and their urine and their sputum, you won't see any of them, because there are so few cells that are required to make you sick, right? So that's a problem with Koch's first postulate. And that's a problem that you have in microbial diseases. When you diagnose diseases, there is sometimes a problem with the sensitivity of the test you're using. And if the test you're using is to look at stuff in blood or urine or sputum, that's a fairly insensitive test. You have to have a lot of microorganisms in order to see them under the microscope, right? So sensitivity, there is a problem with sensitivity. Right, now you might hear in the popular news media today, we're talking about testing for COVID-19 coronavirus, right? And they're talking about, well, we're developing this test and and somebody else is developing another test. And one of the main questions that you ask is, how sensitive is the test? How few viruses can you be infected with? Is there a minimum number of viruses that you have to be infected with before your test is sensitive enough to pick it up? There are lots of tests that are cheap and convenient, but they're not very sensitive. And there are lots of tests that are very sensitive, but they tend to be very expensive. And so one of the tricks that microbiologists have to come up with is they have to develop a test that is both cheap and sensitive. And usually those two things don't come together. Okay, so problem with Koch's postulate number one is that it's a fairly, in order to see it in somebody's bodily fluids, in order to see microbes in somebody's bodily fluids, you have to have quite a lot of them. There are many cases where virulent strains of things are not present in large numbers, and so you will not see them if you just go and look. There, you have to use a more sensitive test. All right, Koch's postulate number two. The suspected pathogen can be isolated and grown in a pure culture. Well, many bacteria can be grown in a bottle, but there are many other microorganisms that can't, because they are something called intracellular parasites. Intracellular parasites, which means that they cannot live outside of a living cell. Right? So there are lots of bacteria that if you just take a bottle and you fill it with water and you add some sugar and you put the bacteria in there, they will grow. But viruses won't, because viruses are only alive if they're inside cells, inside living cells, for example. So we now know that there are many intracellular parasites that need to grow inside cells, and you cannot grow them in a pure culture. You have to grow them in what's known as a mixed culture, meaning that you have to grow up a culture of whatever kind of cells they live inside in order to grow them. So that would be a mixed culture. All right, so the third postulate, if you infect the healthy animals with the pathogen, they must develop the same signs. That's actually pretty good. Number three is a good one. I can't see any flaws with that. Right, and then number four has the same problem that number one has, which is that sensitivity. You have to be able to see it in order to get to number four. So Koch's postulates were pretty good for the 19th century. Today we know that there are cases where it doesn't really work. But anyway, that's that. Okay, now let's move on to the next phase of the lecture, which is discussing the stages of pathogenesis. Right, so how do these microbes get into your body and then cause an infection? It actually has to go through several steps before it can do this. Okay, the steps, in fact, are exposure, adhesion, invasion, infection. Right, so let's have a look at those. Okay, so if you've come into contact with somebody who has an infectious disease, they say that you have been exposed to the microbe. That does not mean that you're infected. Right? So we mentioned in one of the early lectures that if you are exposed to somebody who has tuberculosis, Mycobacterium tuberculosis is a bacterium that causes tuberculosis. If you have come into close contact with somebody that has tuberculosis, you will have to be tested, and if they find out that you've contracted tuberculosis from that person, you have to go into quarantine until you're better. On the other hand, if they test you and you don't have tuberculosis, then they say, fine, you can go. You don't have to go into quarantine, right? So the fact that you were exposed to it doesn't necessarily mean that you were infected. Infection is in fact the final stage. Okay, next, adhesion. Adhesion means that there's something about the bacteria or the virus or the protist that allows it to stick to the inside of your body. Normally, the things that allow it to stick to the inside of your body are virulence factors, things that we classify as virulence factors, right? So we're going to discuss those next. Okay, once it sticks into your body, that it doesn't necessarily mean that it manages to get inside or to hang on when it's inside. Can it survive inside your body? Maybe, maybe not. If it manages to hang on, that's adhesion, and it manages to actually invade your body and survive in there, that's referred to as invasion. But that invasion may only be temporary, right? So your immune system may kill it, or it may just not catch on sufficiently to stay. So it may end up dying out anyway. So invasion does not necessarily mean infection. But if you meet all these conditions, exposure, adhesion, invasion, and it manages to survive inside, then you are infected and you have an infection. Now let's talk about something very important to the pathology and the epidemiology of microbes, and that is the mode of transmission. How does it get from person to person or from animal to person, and how does it get in? Mode of entry, for instance. All right, modes of transmission. Okay, so contact transmission, you know what the word contract means. So contact transmission is from person to person, and contact transmission is sometimes referred to as direct contact transmission or indirect contact transmission. All right, now direct contact transmission is sometimes referred to as skin-to-skin transmission. It's just where you get a disease from another person by actually touching them, handshake, anything from a handshake to sexual activity, very informal contact to very intimate contact is contact transmission. Indirect contact transmission is transmission through fomites, and we already established that fomites are inanimate objects, needles being shared by IV drug users, a pen that you touched and then gave to somebody else, a dirty knife that somebody else got somebody else's blood on it. A doorknob is an easy example of a fomite. Somebody has a cold, they sneeze into their hand, then they turn the doorknob to open the door, then you touch the same doorknob with your hand, and then you wipe a speck of dust out of your eye, you become infected. The doorknob, therefore, was a fomite. Door was a fomite. Vehicle transmission, this term has several meanings, but it refers to as food or water or air or dust, called vehicle transmission, right? So microbes, infectious microbes can be carried in water, they can be carried in food. If you catch a microbe from food or from drinking some contaminated water, you say it's vehicle transmission of the microbe. Vector transmission, we already discussed. It's where the disease, the microbe, the infectious microbe is transferred by an arthropod. Examples of arthropods are insects and arachnids. Most commonly, mosquitoes, insects. The most common insects for this are mosquitoes, and the most common arachnids are ticks, T-I-C-K-S, ticks. Right, those are the common arachnids. All right, let's look at contact transmission in greater detail. Direct contact transmission means person to person, touching, kissing, etc. Right, direct contact transmission. Now here's a very important term you must learn: droplet transmission. Droplet transmission refers to sneezing on people. When you sneeze, droplets of saliva and sometimes mucus come flying out of your mouth and your nose. They generally have a range of between 0.5 meters to 1 meter, right? And the reason you've heard in the social media all over the place about social distancing being 2 meters. Why do we say that? Because the range of droplet transmission is typically 1 meter, right? So if we're maintaining social distancing of 2 meters, that means somebody can sneeze when they're 2 meters away from you, and you will probably not get infected. Probably. Not necessarily, but probably not. Right. Another way that you can minimize the effect of being infected by droplet transmission is to wear a mask. And so if you wear a mask, it's not to protect you from getting infected necessarily, although it does sort of do that. But the main reason for wearing a mask is that if you happen to be infected, if you have a disease and you sneeze, you won't transmit it to other people through your droplets. By the way, a very important definition here is that a droplet is defined as a drop of bodily fluid, saliva or mucus that is larger than 1 micrometer in diameter. If you have a droplet of bodily fluid that's smaller than a micrometer, that's referred to as an aerosol. An aerosol. So there is droplet transmission of disease and there is aerosol transmission of disease. Typically when you exhale, mist comes out of your lungs, a little bit of mist, and that is aerosol transmission. The range for aerosol transmission of infectious microorganisms is usually about only 10 centimeters or so. So the distance for transmission of aerosols from person to person is only about 10 centimeters versus droplet transmission, which is 1 meter. So your odds of being contaminated by somebody sneezing are much greater than being contaminated by somebody just breathing in front of you. However, there is evidence that if you have people in a confined space, the aerosols, the water droplets from the aerosols will build up to a point where it is possible to infect somebody. If you just have 2 people in a confined space that are breathing the air, eventually the aerosol concentration will become high enough that you could infect somebody with aerosols. So that's why throughout this coronavirus epidemic, this coronavirus pandemic, we're recommending that if you drive with somebody in a car, you roll down the windows. That's to avoid buildup of aerosols to the point where you can infect somebody. So you're supposed to maintain social distance, and you should also keep the air as fresh as possible. You need to keep rooms and vehicles ventilated in order to prevent having a buildup of aerosols to potentially cause aerosol transmission. All right, now there's a difference between a vertical direct transmission and horizontal direct transmission. Vertical, of course, means from top to bottom, right? And vertical contact transmission, or just plain vertical transmission, refers to the transmission of a disease from a mother to a child during pregnancy. Those types of transmissions are also referred to as transplacental infections. Transplacental infections. And there are a few microbes that can cause transplacental infections. Most of them are not. Most of them will not cause transplacental infection, but some do. Horizontal transmission simply means from between person to person, 2 people that are not related or whatever. Vertical transmission specifically refers to mother to child transmission, specifically during pregnancy. All right, indirect contact transmission, of course, means through fomites. Right? So droplet transmission is not considered to be fomite transmission. It is considered to be direct transmission. So droplet transmission is defined as direct transmission. Fomite transmission through inanimate objects like doorknobs and money and railings on staircases is referred to as indirect contact transmission. So droplet transmission, the range is about 1 meter. We ask for social distancing during pandemics. If you have a disease that is being spread through droplet transmission, the coronavirus is spread through droplet transmission, and that's why we talk about social distancing of 2 meters, because that's generally twice the distance of droplet transmission when somebody sneezes or coughs. You even get droplets forming when people are just speaking, right? So if you've ever watched somebody speak when their face is silhouetted against a strong light, like somebody's face is silhouetted against a beam of sunlight, just speaking, you'll see this kind of spray coming out of their mouth, and they're not particularly juicy speakers, but you'll see droplets come out if the light is strong enough. So this image on the bottom left is not doctored in any way. It's not photoshopped. This is actually the droplets that come out of somebody's mouth when they sneeze, and you can see right there that they have a range of about 1 meter. A few of the droplets can travel farther than that, but it's rare for any droplets to travel farther than 2 meters, and so that's why we ask for a social distancing of 2 meters. Now the image on the upper right is of a bunch of people riding the subway, some of whom are wearing masks. Now this is not a modern picture. This photograph is about 10 years old. And those of you who are from Asia, Japan, South Korea, China, you're used to seeing people doing this on the subway. And this was common in Asian countries long before the corona pandemic. And these people are, so this is an old picture from 2010 or something like that. These people are, some of the people are wearing masks and some of them are not. The people who are wearing masks are people who are going to work sick. They're going to work with a cold. The anthropologists will tell you that North America and Asia are what we call work while sick countries, which means that there's so much pressure on the workers that they either feel that they have to come into work even though they're sick, or their boss or their employer tells them that they must come to work when they're sick, or they're fired. You're fired if you don't come to work. You're missing or just simply by the fact that many employers don't offer their employees sick days, which means if you stay at home when you're sick, you lose money, which you may not be able to afford to do. So we are a work while sick culture. So ideally, you should not come to work when you're sick, because then you don't spread the illness. But if you must come to work when you're sick, if it's a contagious disease that's spread by droplets, you wear a mask. So if you have influenza or a cold, you wear a mask on the way to work. In North America, this was not a common thing until the corona pandemic. Hopefully, they talk about every cloud having a silver lining. Cloud having a silver lining, I hope, from the coronavirus pandemic is that the entire world learns that it's a good thing to wear a mask, right? It's a good thing to wear a mask if you're sick. So these people are wearing masks not to protect themselves from getting infected. They are wearing the masks as a courtesy to their fellow citizens, to avoid infecting their fellow travelers, to avoid infecting the passengers. So you see, here's a man who's obviously not sick. These people are sick, so they wear a mask. So in North America, we have a very big thing against smoking. There's some countries in the world where a lot of people smoke. In North America, we try to discourage it. Lots of people smoke anyway, but we try to discourage it. It's looked on as kind of a social taboo. So if you saw somebody riding the bus smoking, everybody would be staring at them and giving them nasty looks like, you're a bad person for smoking in here because we're inhaling your secondary smoke and you're giving us lung cancer. Well, in Asia, if you get onto a bus and you're coughing and sneezing without a mask, people give you the same looks like, what are you doing? You're making us sick. Why aren't you wearing a mask? So it's a common thing to wear a mask if you're sick in Asia, and we need to make that a common thing in North America and South America and Europe and everywhere else. It should become the normal thing to do, that if you must go to work, you wear a mask, and people don't make fun of you for wearing a mask, or people don't stare at you for wearing a mask. People smile at you for wearing a mask and say, thank you for not spreading your germs to me. Thank you for taking precautions for not spreading your germs to me. All right, so some of the professions, it's very common to wear masks. So on the left, we see a dentist. And so notice that she's wearing eye protection and a mask, right? So the eye protection is to prevent her patient from coughing into her face and infecting her, and the mask is to prevent her from coughing into her patient's face and infecting her patient, right? And here we have surgery. Of course, people are wearing a lot of what's called PPE, or personal protective equipment, but in this case, it is, the person is on an operating table. They're not going to sneeze in your face. All of this equipment, this protective equipment that they're wearing, is exclusively for the benefit of the patient, to prevent any of the microbes that these doctors have in their body from infecting the patient. So they're wearing goggles to prevent tears from dropping into the patient and possibly carrying diseases. They're wearing a hair covering to prevent dandruff flakes from falling into the patient and causing illnesses. They're wearing a mask to prevent droplets from falling into the patient and causing diseases. They're wearing gloves to prevent microbes on their skin from getting into the patient and causing an opportunistic infection. And they're wearing a gown. They're wearing gowns to prevent any microbes or usually fungi or yeast that are clinging to their clothes from infecting the patient as well. So all of these things are meant to prevent the normal human microbiota from infecting the patient on the table. If you've been to restaurants, you notice that in most places in the world, it is the law for a salad bar or a buffet to have a sneeze guard. The purpose of the sneeze guard is to prevent droplets from the customers from getting into the food. And so in most places in the world, it is required that you have a sneeze guard. Most people here are taller than the sneeze guard, so there's no possibility. But if you have children, or if you have people who are shorter, you notice that there's some ladies on the side over there that are fairly short. I'm sure that when they're talking, many of their droplets are getting underneath the sneeze guard. So the sneeze guard, this sneeze guard is obviously designed a little bit imperfectly. It should, ideally, a sneeze guard should be low enough that just your hands can fit under. So just your hands and your plate should be able to fit underneath the sneeze guard. This one is actually too high. Okay, so this is something that you would worry about if you were a restaurant inspector, right? So if you go to BCIT, British Columbia Institute of Technology, they offer a program to train you to be a government inspector. It doesn't guarantee that you'll get a job with the government being an inspector, but at least you'll have the certificate so you can become an inspector. That's one of the jobs that you can get if you're a microbiologist. And some of you may know Dr. Baines, Onkar Baines, from the biology department here at Columbia. He used to teach in that program. It's called the Environmental Hygiene Program at BCIT. So if you're interested in that program, you could talk to him about it. And that's one of the things that you would be looking for if you were a restaurant inspector. If you were a health inspector, you'd go into the kitchen and you'd inspect a bunch of things, and then you would come out and you would say that you need to, it's the law that you have to put a sneeze guard above the salad buffet or the salad bar, and it has to be lower than this. Okay, so these are the methods of transmission of infectious microbes. Now let's look again at vertical and indirect contact transmission. Okay, so vertical direct contact transmission referring to mother to child. Notice here, notice that I'm sure you all know that the baby doesn't breathe because it's in the mother's womb. Its blood becomes oxygenated with the help of a structure called the placenta. The placenta is a space where the mother's blood vessels and the baby's blood vessels come close enough together that oxygen can be transferred to the baby's blood and carbon dioxide can come out of the baby's blood back into the mother's blood. And the placenta filters out a lot of diseases from the mother's blood. So there are a lot of bacterial diseases. If the mother gets bacteremia or septicemia, the baby will probably not have any problem because the placenta is actually a good filter. The placenta filters out a lot of problems so that they do not infect the baby. However, there are some infectious agents that can go through the placenta, and we call those transplacental infections. Okay, here what you're seeing over here is a mosquito biting the mother. And that's inferring, they're trying to suggest there that the baby is going to catch Zika virus. You may remember a few years ago, we had an outbreak in South America and Central America of a disease called Zika virus, which was carried by mosquitoes, carried by a mosquito vector, which caused the babies to be born with a lot of problems, including mental retardation and microcephaly. Microcephaly means abnormally small head and a small brain, which leads to mental retardation. So Zika virus is an example of a transplacental infection. Now notice down here below the picture, I've written TORCH, torch. Torch is an acronym. An acronym, of course, is a word that's made up of the first letters of a bunch of other words. TORCH is an acronym that stands for a bunch of diseases that can be transmitted transplacentally. The T, for example, do you remember from the first lecture, the lecture number 1, what is the name of the protozoa that's carried by domestic house cats that can cause birth defects if a pregnant woman catches it? The microbe is called, do you remember? It'll be on the tests. The microbe is called Toxoplasma gondii, and the disease is called toxoplasmosis. And toxoplasmosis usually causes subclinical infections in adults, asymptomatic infections in adults, which means that if an adult has toxoplasmosis, they usually don't know because they don't feel sick at all. But if it's a pregnant woman, the Toxoplasma gondii can get through the placenta, transplacental infection, and cause birth defects in the baby. Now, what about indirect contact transmission through fomites? These are all typical fomites. A doorknob is a notorious fomite. Money is notoriously dirty because everybody is touching it. Coins, you can sanitize them, so you can dump all of your coins into the water and wash them, or better yet, just when you handle the coins, just don't touch your face, because most infections get in through the face, particularly through the eyes. Money, some countries in the world use paper money, and some countries use plastic money. It looks like it's paper, but it's actually nylon or something like that. Now, the plastic paper money is, you can wash it, and so it can be cleaned periodically. Paper money is notoriously bad because you can't wash it, and paper often becomes contaminated with fungus because fungi, like the fungi, if you own a bookstore, the books will often become contaminated with fungus because paper is made out of cellulose, and fungi are some of the very few organisms on Earth that can actually digest cellulose. So paper is notorious for getting contaminated by fungal infections, fungus infections. And so librarians have to know all about controlling the humidity in the library, because if the books become damp, they will get fungal infections, which is called book rot. And so you often walk into bookstores and you suddenly feel your eyes start to itch. You walk into a badly run bookstore where they sell used books, not a new bookstore, but a used bookstore. You walk in, this happens to me all the time. I walk into a used bookstore and immediately my eyes start to itch, and I know that my eyes start to itch and my throat starts to hurt and feel scratchy. And I know immediately that those people that own that used bookstore have not been controlling the humidity, and the books are growing mold. And so you get out of there, because mold is actually very bad for you. All right, so if you're a librarian, you have to know all about, or if you own a bookstore, you have to know all about keeping it as dry as possible in there to avoid growth of fungi. And money that is made of, if you have paper money, that is susceptible to fungal infections. Okay, now look at this guy on the bottom right, gripping the bars on the bus, and the handrails on staircases are notoriously filthy. They are perfect fomites because everybody touches them, right? So fomites are notorious for transmitting diseases from person to person. As I said, intravenous needles are also considered to be fomites. So intravenous needles are shared sometimes by drug addicts. All right, now let's just talk a little bit about microbial pathology and public policy, public healthcare policy, because that's an important part of infectious diseases. Right now, what do you do with drug addicts who share ultraviolet intravenous needles? Okay, now first of all, some people dislike or feel contempt for drug addicts. Personally, I think that's a very bad attitude, because if you think that these are bad people because they have a drug addiction, probably means you have no idea how addictive these drugs are. Anybody who's ever tried to quit cigarette smoking and they've had so much difficulty with it, you have regular people who smoke cigarettes and they say, I want to give this up, and they try and they try and they can't. Drug addiction is 1,000 times more potent than cigarette addiction. It's 1,000 times stronger. People who they make a mistake at one point in their life, they try it once and then they get addicted, or they try it twice and then they become addicted. And from then on, the trajectory of your life changes completely, and you're not in control anymore. The drugs are. Basically, your brain will do anything to get more drugs. And drug addicts will often spend what little money they have on drugs rather than food. So if you look at drug addicts, they're often starving because they don't even care about eating anymore. They will spend every penny they have getting drugs, right? And so the urge to continue using these drugs is so strong that there's almost nothing you can do to stop it. Okay, so the people who are addicted to drugs are not contemptible people, and I know firsthand that many of them, when they recover, they were wonderful people before they had an addiction. They become terrible people when they're addicted. once they recover again, if they're able to recover, they become wonderful people again, and everyone is somebody's son or daughter or brother or sister or father or mother. There are people that love them. They don't deserve to die. They don't deserve to catch these diseases. So, and even if you don't accept that argument, another argument is that Canada, like most countries, has a public health care system, and so that if these drug addicts contract hepatitis C from a fomite, like a needle, there's a whole other set of health problems that are associated with hepatitis. So hepatitis is commonly transmitted. Both hepatitis is caused by 3 different viruses. Hepatitis and HIV/AIDS are all transmitted by drug users sharing intravenous needles, right? So even if you don't believe, even if you don't like these people, we still have to pay for their health problems if they become sick. And so a public policy solution, which nobody likes particularly, but it's a solution that works. So if you go into public policy, many of you will, I hope, public policy sometimes has to be crafted in a way that nobody is happy, but it's effective. It's for the greater good. No public policy is pure good. Most public policy is a compromise that has some bad things about it, but the good things greatly outweigh the bad things. Okay, now Vancouver, every country has dirty little secrets that they don't want foreigners to know about. Some of you come from other countries that have bad things that happen that you're kind of embarrassed about, and you don't like other people to know that they're going on. Okay, so Canada has dirty little secrets as well. We have secrets that we don't like to talk about. We have bad things that go on. Canada is generally a very good country, but there's some bad things that happen that we don't like to talk about, and if we don't talk about it, that the problem will go away if we ignore it, and it doesn't. Right? So one of Canada's dirty little secrets, Vancouver's dirty little secret, is that Vancouver happens to be the drug use capital, the intravenous drug use capital of Canada. Not only that, but downtown Vancouver, specifically at Hastings and Main, in Vancouver, is the intravenous drug abuse capital of Canada. Right? So there are more drug abusers in downtown Vancouver near Hastings and Main than anywhere else in Canada, and indeed more IV drug users there than many countries. And the reasons for that are not clear. The anthropology teachers at the college might give you different answers. One of the theories is that most of Canada is so cold that it freezes in the winter, and because drug addicts will spend all of their money on drugs and very little of it on housing, many drug addicts are homeless. And if you live anywhere but Vancouver in the wintertime, you'll freeze. If you live out on the street in a tent, you'll freeze. And so they tend to end up in Vancouver because it's warm enough to survive. A few of them end up in Victoria as well for the same reason. So Victoria and Vancouver are the 2 cities in Canada that rarely freeze. They rarely go below 0 in the winter. So that's one theory. Another theory is that Vancouver is a port city, and many of these drugs are smuggled into the country on ships, and like any other an economist will tell you that things are goods, including illegal goods, are cheaper at the source. So if we have all of these drugs coming into the port of Vancouver on ships, it stands to reason that the drugs would be cheaper here. And so intravenous drugs are cheaper in Vancouver than they are anywhere else. Why can't we arrest the drug dealers? Well, they are trying to do that. Can we arrest the drug addicts? Well, not currently, because it's not a crime to be addicted to drugs. There are people that are occasionally drug addicts because they often steal things they steal things and then sell them and use the money to buy drugs. They tend to be behind a lot of crime, and so some of the judges will say, well, I'm convicting you of stealing, but I'll give you a choice between going to jail or going into a drug treatment program. And so the law, you can kind of tinker around with the law that way to encourage people to get into rehabilitation programs to get rid of addictions, and that's one way to get people into it. But being a drug addict itself is not against the law, but because many drug addicts steal things in order to support their drug habit, you can sort of use the law in order to cure some of these people. But back to the original point, one of the solutions that we have found in Canada, it's based on a solution that they found in Holland. In Holland, they have drug addicts as well, and they started out by introducing what are known as safe injection sites. So in Vancouver, Vancouver was originally the first city in Canada to try the concept of safe injection sites to lower the transmission of hepatitis and AIDS, HIV, by giving drug addicts clean needles. Now, what happens is that we have 3 of these sites in Vancouver. One of them right downtown. It's only about 5 blocks away from Columbia College. Where if you're a drug addict, they will not sell you drugs, right? So there's a misconception that they sell drugs or they give you drugs. They don't. But if you're a drug addict, you buy drugs from criminals that are selling drugs, right? Okay, so you get your drugs from a criminal, but if you then take those drugs into the safe injection site, there is a nurse there that will give you a clean needle and will offer you some food, because drug addicts are often starving. And the nurse will also offer you some advice on ways that you can possibly go into treatment programs to avoid the addiction. Right. So if you go into this safe injection site, they'll give you some food, they'll give you a clean needle to inject your own drugs, and if you're willing to receive it, they will give you some advice. And so we have actually, once we introduce these safe injection sites, first of all, when drug addicts are injecting drugs, they often overdose. And if they inject drugs in an alley, which is a popular place to inject drugs, you know, behind the big dumpsters in the alley downtown or something like that, many times they will overdose, and sometimes they'll die. Even if they overdose, they have to, and if they're rescued and saved, they have to send paramedics with an ambulance and everything, and it's very expensive to rescue them. So if you overdose inside the safe injection site, there's a nurse there already, and the nurse will resuscitate you, right? So as soon as Vancouver introduced the safe injection sites, the frequency of people dying from overdoses dropped by 90%. Right? So 90% of the drug overdose deaths were prevented by the introduction of the safe injection sites. Right? So that's already a very good benefit in itself. In my opinion, that's one very good benefit. Secondly, the rates of hepatitis B, hepatitis C, and HIV have gone down somewhat because there are fewer people who are spreading these diseases by sharing needles, right? So it does have positive benefits. There are people that I think are mistaken, but there are people who claim that, well, you're making it easy for people to stay addicted to drugs by giving them a clean, comfortable place to inject their drugs. And if you force them to inject drugs in the alley with dirty needles, they might be so afraid of getting hepatitis that they won't be a drug addict anymore. Frankly, anybody who says that is a fool, and they don't know how addictive these drugs are, right? So if people would rather spend their money on drugs than on food, so they're literally starving to death, the point where teeth are falling out of their head because their proteins are breaking down, forcing them to inject those drugs in the alley is not going to discourage them from injecting those drugs. So think of that, and you'll realize that that argument is silly, saying that if you give these people a safe place to inject drugs and a clean needle, that's just encouraging drug use. And versus if you force them to inject drugs in a dirty place with a dirty needle, there will be fewer addicts. That's not true, right? So that is a foolish statement, in fact. Okay, so in Vancouver, we injected 3 of these safe injection sites specifically to prevent the spread of hepatitis and the spread of HIV through dirty needles as fomites. Right, so on the upper left, we have the inside of the safe injection site. You can see way at the back, there's a nurse who's passing out clean needles. Everyone injects their drugs like it's like a library booth or something like that. The alternative to that is shown on the bottom right, where people inject drugs in the alleys in downtown Vancouver, and the paramedics have to come and resuscitate people. It's a mess. It's a terrible thing, and the safe injection site is solving this problem little by little, slowly. There may be faster ways to solve it, but we've never come across any. So the safe injection sites are working slowly. Their use is controversial. Now you're aware of it. You know the facts. Knowing the facts is always a good thing. Knowing accurate knowledge is always a good thing. So here I've told you the facts about the safe injection sites. Maybe if some of you are studying anthropology, you want to talk to your anthropology teacher about studying the anthropology or the sociology of drug addiction and various methods of dealing with it, and this is one of them. So this is a case study in the sociology of drug addiction, and also a case study in public policy about how to avoid drug addiction. Okay, so all of that came from our discussion of fomites. This is one of the dirty needles, dirty intravenous needles being shared by drug addicts is one of the most dramatic examples of diseases, serious diseases, highly lethal diseases being spread by fomites. Vector transmission. Okay, so there is a difference between a biological vector and a mechanical vector. A biological vector, when we talk about a vector, we're usually talking about an insect or an arachnid. A biological vector is where the vector itself is infected, right? So in the case of malaria, the mosquitoes can carry, do you remember what the reservoir is for malaria? So humans get it from mosquitoes. Humans do not give malaria to each other directly through contact transmission or anything like that. A human can only get malaria from a mosquito. Right now, where do the mosquitoes get it? They get it either from other humans, or what was the non-human reservoir again? Do you remember? It'll be on the tests, I guarantee. The non-human reservoir for malaria is monkeys. Right? So now the mosquito carries the malaria from person to person, or from monkey to person, or from person to monkey. Indeed, from person to monkey is possible. And the mosquito itself is actually infected, which means that the malaria parasite, the Plasmodium vivax, the Plasmodia, can live inside the mosquito. In fact, the mosquito is actually what's known as the primary host for the malaria parasite. Now, that brings up another definition. What is the difference between a primary and an intermediate host for a parasite, or a protozoa in this case? Okay, so the protozoa that's causing malaria is Plasmodium vivax, Plasmodium. And protists often have a number of different hosts that they can live in, and they have a number of very different odd forms that they can exist in, and we'll talk about that later. The primary host for that protozoa is the host that allows those protozoa to go through all of the different stages of their life cycle inside that particular host. An intermediate host, which I will sometimes refer to as a secondary host, but the correct term is actually intermediate host. An intermediate host is a host for that protist that does not support all of the different forms that protist can exist in. The most significant one is the sexually reproductive stage, right? So Plasmodium vivax, for instance, has an asexual stage where it just reproduces by binary fission, by splitting in half. And it has a sexual phase where there's a male and a female version of it, right? So the male and the female version of it cannot live inside humans or monkeys, but it can live inside mosquitoes. And so therefore we call the mosquito the primary host for Plasmodium vivax. The humans and the monkeys are the intermediate hosts, right? So in fact, if you got rid of the mosquitoes, malaria would eventually die out, because humans and monkeys can't support sexual reproduction of malaria. So in fact, most of the countries that have malaria target their efforts to get rid of malaria by targeting the mosquitoes, but they've never actually succeeded, because there's so many mosquitoes that you can't possibly get rid of them all. But that's usually what they're trying to do. They don't go around trying to kill all the monkeys. They go around trying to kill all the mosquitoes. Monkeys are cute. A lot of people object to killing monkeys, but they do carry a lot of diseases. So even though monkeys are cute, I would not advise having them as pets, because many diseases are spread from monkeys to humans, and the reason for that is because humans and monkeys are very similar. So any disease that a monkey can catch is it's usually possible for a human to catch it. So I do not advise people being around monkeys. Humans should avoid monkeys. But nevertheless, so we're talking here about malaria. Mosquitoes are the primary host for malaria. I will probably ask you that on a test at some point. If you got rid of the primary host, the disease would eventually die because the intermediate host can't support it. Okay, what's the difference between a biological vector and a mechanical vector? Mechanical vector is what we call an insect or an arachnid that carries the disease from one place to another, but it is not itself infected. Including, we discussed the fact that anthrax is carried by animals, hooved animals like cows, sheep, and horses, primarily sheep. But they are not themselves, usually are not themselves infected with it. They just carry it in their fur. They carry the anthrax microspores and the endospores in the fur. If you have a fly, an insect, you have a fly that lands on excrement, which they often do. Black flies do that all the time. And then they go and they land on your food. They transmit some E. coli from the excrement to your food, and then you eat it. But the the fly itself is not contaminated with E. coli. Then you call the fly a mechanical vector. So here we have a fly landing on some excrement over here. Disgusting, but that's often what flies do. And then they fly over here and they land on your hamburger bun, and then you eat the hamburger and you become infected. The fly was technically classified as a mechanical vector. Versus a mosquito biting a person and giving that person malaria. The mosquito is a biological vector because the mosquito itself is infected. The mosquitoes are, we don't know whether they're miserable or not, but because of the infection, but they are in fact infected by the Plasmodium vivax. All right, let's talk about vehicle transmission. So air, dust, water, and food are all considered to be vehicles for the transmission of disease. Okay, so dirty air, dirty ventilators. Dust is very common. So up here on the top right, we have a woman sweeping up dust. Dust is particularly bad because dust often contains the skeletons of arthropods like dust mites, and more importantly, it often contains bird droppings, and bird droppings often contain fungal spores, and fungal spores are particularly bad because if you inhale them, then you get diseases that are called mycoses that can spread from your lungs into your blood and cause a systemic mycosis. Food, of course, down on the bottom left, you know that Clostridium botulinum is transmitted through a food vehicle. Water vehicles, including ice cubes, of course, can be a transmission of disease. In the case of food and water, the disease gets in through what we call the oral fecal route. The oral fecal route, which means that oral meaning you eat it. All right, portals of entry. Let's talk about that. Okay, so first of all, the mucous membranes, any body tissue that produces mucus, including the eyes, the nose, the vagina, the urethra, the penis, and the rectum, any of those things are often infected by microbes because many microbes will stick to mucus and then get inside. So we talked about attachment factors, adhesion factors. Many of them will stick to mucus. I mentioned earlier that the parenteral route, very important term, I'll probably ask you that on a test, means a break in the skin, a cut, a needle, a puncture. So transmitting hepatitis by infected needles, the hepatitis gets into the body via the parenteral route. Hepatitis gets into the body via the parenteral route. The oral fecal route means eating contaminated stuff, contaminated food or contaminated water. In the case of water as a vehicle, contaminated water is usually part of what's known as the oral fecal route for infection because it usually happens where water is contaminated with small amounts of sewage. So if you have cities or villages that are dumping sewage directly into a river or a body of water, it stands to reason that, and there are other people who are using that body of water for drinking water, Sometimes people will say, well, it doesn't matter because the amount of sewage that gets into that water is very small compared to the actual volume of the water. Well, we already know now that there are virulent strains of bacteria and other microorganisms that are virulent in very small numbers. And that's usually what happens. And so the oral fecal route usually means that somebody drinks water that is contaminated by a small amount of sewage that came from another human being that was contaminated by a disease. Right? So Vibrio cholera, we'll talk about that later. Vibrio cholera is a bacteria that gets into water supplies that are contaminated with sewage, and so you have a person who's got cholera. A little bit of their excrement gets into the water supply, then a lot of people drink it, and then they get cholera as well, and then they excrete, and if any of their sewage gets into the water supply, then we've perpetuated the cholera. We've perpetuated the circle. All right, remember the parenteral route. Okay, so here are some of the roots. Okay, so the eyes and the conjunctiva and the nose and the mouth are examples of mucous membranes, right? The placenta is responsible for transplacental infections. Things can get in through the vagina, the urethra, breaks in the skin, the parenteral route. Insect bites, example of parenteral. Needles, example of parenteral infection. How do they get out? Okay, so egress means to go out. So you have viral egress and bacterial egress through any of these bodily fluids, including tears, earwax, mucus. So if you have a cold and you're sneezing, you're sending droplets of saliva and mucus flying all over the place. Mucus, saliva, breast milk. Some diseases can be spread through breast milk. In fact, we're going to talk about a common disease called brucellosis, which is transmitted from cow's milk, and occasionally you can get different types of infections through breast milk of any animal, but also human breast milk. Transplacental infections we talked about transmitting from through the placenta. And then vaginal and penile secretions during sex. Those are common ways to transmit diseases. Broken skin, blood, IV use, and so on, and up to and including skin flakes, dandruff. So it's a bad idea to be in contact with other people's dandruff. One of the interesting things about dandruff, we notice dandruff because the skin is flaking off underneath our hair from our scalp. And then the flakes of dead skin get trapped in our hair and you can see it. But the same thing is actually happening all day long to all the other parts of our body. You just don't see it because it doesn't get caught in our hair. It also happens when we're asleep, which means that flakes of human skin are constantly being shed into the blankets and the sheets that we sleep in. And if you go long enough without changing the sheet, the flakes of skin will actually make their way into the mattress. So it's a good idea. It's a good idea to wash the blankets and sheets regularly, as often as possible, to avoid accumulation of skin flakes. And more importantly, if you're the owner of a motel or something like that, that's the reason why you have to wash the sheets all the time. You have to wash the sheets every day and the blankets every day. But more importantly, you have to change the sheets regularly to prevent flakes of skin from getting from the sheets to the mattress. And then you have to get rid of the mattresses. You have to replace the mattresses every couple of years because they become saturated with skin flakes. Right? So diseases, not only can diseases be carried in skin flakes, but also a lot of insects, microscopic insects, dust mites, like to eat the dead skin flakes, and so that's why you get these mites inside a mattress. If a mattress makes you sneeze, it's probably because of the dust mites. The exoskeletons of the dust mites are very allergenic, so the shell of those mites, microscopic mites, if you inhale those, they generally make you sneeze quite violently because they're highly allergenic. Okay, so there are some of the portals of exit. Transplacental, the placenta filters out a number of diseases, but not all of them. I will ask you about this on tests. In order to keep track of which diseases can be transmitted through vertical transmission, transplacental transmission, we've come up with the acronym TORCH, T-O-R-C-H. We'll learn what a few of them mean. Okay, so there's a transplacental infection. TORCH stands for toxoplasmosis. The T stands for toxoplasmosis, meaning Toxoplasma gondii. The R stands for rubella. Remember congenital rubella syndrome. C stands for cytomegalovirus, specifically a virus called human herpes virus 5, cytomegalovirus. And then another herpes virus, HHV2, human herpes virus 2. So that's kind of cheating, right? We have two herpes viruses, but we give them two different letters. So C stands for cytomegalovirus, which is a member of the herpes viridae family, and herpes, which is an example of the herpes viridae family. And then again, we're going to kind of cheat and use O for other, and the other includes syphilis, chickenpox. Chickenpox is Varicella-Zoster, hepatitis B, and HIV, the AIDS virus, right? So remember that syphilis, chickenpox, hepatitis B, and AIDS can all be transmitted from mother to child during pregnancy, right? As well as toxoplasmosis, rubella, cytomegalovirus, and herpes virus. So at some point I will probably ask you on a test multiple choice questions about what the letters in the TORCH acronym stand for. I just want to tell you briefly what a teratogen is. You need to be familiar with this word. The word teratos is an ancient Greek word meaning monster. And the word gen means to create. So teratogen specifically refers to the source or the cause of monsters being born. That's not a very politically correct word, but what it means is that there are things that can happen to a mother, or a pregnant woman can be exposed to certain things that cause her baby to have birth defects. And then monstrous birth defects, I guess, is what they were trying to say, but that's obviously a very negative term, but that's still the one we use. All right, so teratogen is something that causes birth defects, and there's a difference between a physical teratogen and a chemical teratogen and a biological teratogen. Okay, so physical teratogens mean if you have a pregnant woman and she's exposed to radiation, or she gets into an automobile accident so that her belly is hit by the steering wheel or something like that, any of those things could cause deformities in the developing child who's developing in the womb. Some chemicals are considered teratogens, alcohol for instance. So you've heard of fetal alcohol syndrome. That's something that happens if the mother drinks too much alcohol while she's pregnant. The baby can be born with a syndrome called fetal alcohol syndrome. There was a chemical about 60 years ago that was put on the market. It was a sedative called thalidomide. That drug was not properly tested before they sent it to market. And what happened was that drug turned out to be a chemical teratogen that caused thousands and thousands of children to be born without arms and legs. It was a terrible thing. It happened in about 1961-62 before they figured out what was going on. The epidemiologists looked at the women who were giving birth to children that had no arms and legs, and they found out that the one thing that they all had in common was that their doctors had prescribed them all thalidomide as a sedative because they were all nervous for some reason, and they all took thalidomide, and they were all born with children. All of their children were born without arms and legs, and so the epidemiologists were able to determine that the one thing they all had in common was that they all had thalidomide, and so now we know that. That's why it's very important to test, make sure that drugs are thoroughly tested before they are given to the public. Obviously, they weren't in that case. All right, and finally, the reason for us being here today, talking about biological microbial things that cause birth defects. These are classified as biological teratogens. Example, syphilis. Syphilis causes bone deformities if a pregnant woman catches syphilis during her pregnancy. If a pregnant woman catches rubella during pregnancy, the baby will have congenital rubella syndrome. Or if a woman contracts toxoplasmosis by cleaning the cat litter box or something like that during pregnancy, there are various birth defects associated with that as well. Example of a biological teratogen, congenital rubella syndrome caused by the Rubella virus, causes a disease known as the German measles. Okay, what are the symptoms and signs of congenital rubella syndrome? The baby has a small head and mental retardation. Small head is called microcephaly. The word cephalus refers to the head, so microcephaly literally means a small head. If you have a smaller brain, that generally leads to retardation as well, but not always. Problems with the eyes, cataracts on the eyes causing vision problems, and problems with the heart. Specifically, I will not ask you about this, but coarctation of the aorta. Those of you that took the anatomy course here at Columbia know what that is. The aorta is the large artery that comes out of the heart, and if it's pinched in the middle, that's called coarctation of the aorta, which causes a little bit of heart problem. And so that's one of the things that that's one of the characteristics of congenital rubella syndrome. Right, so here's a baby born with congenital rubella syndrome. It has a small head and mental retardation. It has coarctation of the aorta, a heart problem, and has vision problems due to cataracts. It's very, very tragic, and it can easily be avoided if the mother had had a rubella vaccination before becoming pregnant. If you get the vaccination during pregnancy, it's too late, and the vaccine itself may actually cause congenital rubella syndrome. So it's critical for a woman who's planning on becoming pregnant to be vaccinated against rubella long before becoming pregnant. In most countries, that's not a problem because most countries give all the children, or as many as they can get to, the measles, mumps, and rubella vaccine, the MMR vaccine, at 13 months of age, so that they will not, it won't be an issue, right? So the woman won't, by the time she's old enough to get pregnant, she won't get rubella because she was vaccinated when she was 13 months old. All right, let's move on to our discussion of what is it that makes certain things worse than others. What is it that makes certain microbes worse than others? And those things we call virulence factors. Things that make a microbe more virulent. We already determined how we decide how virulent something is. We do experiments and determine the ID50 or the LD50. Things that have a low LD50 and a low ID50 tend to be very virulent. Things that have a high LD50 or high ID50 are less virulent. Okay, now what types of things would make something more virulent? Okay, so number one, adhesion factors are things that allow the microbe to stick more easily. Exoenzymes are usually, the word exo means out, and exoenzymes are enzymes that are sent outside of the bacteria or the protist or the yeast, usually that kind of dissolve or break down the tissue in the surrounding area, which allows the bacteria or the fungus to spread more easily. Exotoxins are literally poisons that are sent out by the bacteria or the fungus that poison you, and so things that produce poisons tend to be deadly in smaller numbers than things that are not poisonous. Endotoxins, the word endo means inside, the word exo means outside, so the word endotoxin refers to toxins that are produced and are kept inside the cell unless you kill the cell, and which allows them to become released. Okay, so do you remember about 20 minutes ago what we were talking about when we said that? Gram-negative bacteria are famous for having an endotoxin called lipopolysaccharide, and the O antigen, we'll talk about that in a minute, but they're famous for having endotoxins that if they infect your blood and you kill them with antibiotics, they may very well kill you, because when they die, they release the endotoxin. Okay, now another thing that makes things particularly virulent that we're going to talk about are ways to avoid getting killed by the immune system. So if you're infected by something, if you're infected by something, your immune system will usually kill it. One of those microbes will be particularly virulent if it has some sneaky, tricky way to avoid being killed by your immune system. And these things here at the bottom of our list are ways that various microbes can avoid being killed by your immune system. If they can avoid being killed by your immune system, they tend to be more virulent than things that can't avoid that. Adhesion factors. In general, two things that make it easier for a bacteria to stick to your cells are, number one, fimbria. Fimbria is a fancy word for hairs. So there's some bacteria that appear to be hairy. These little hairs that are covering the bacteria are called fimbria. right? There are some fimbria that are made of a protein that allows them to stick to human cells more easily than if they didn't have those hairs, right? And another thing that you find often sticking out of a bacteria is called a pilus. A pilus. The word pilus is a Latin word that the ancient Romans used to speak. The language Latin was spoken by the ancient Romans, and in the field of biology, you hear a lot of Roman words, a lot of Latin words, and a lot of Roman words. Not because the Romans invented microbiology, but because microbiology became a science during the era when everyone expected academics to be able to speak Latin. So if you couldn't speak Latin, you weren't considered to be a learned academic. So they named a lot of things. A lot of these things have names that come from Latin and Greek words, ancient Greek words. And the word pilus is a Latin word meaning spear. It literally translates as spear, that the Roman soldiers used to throw at each other. Now, some bacteria produce a pilus that they use to spear other bacteria during a type of mating called conjugation, which we'll talk about later. But that same spear, that pilus, can also be used to attach to host cells. It can be used to attach to human cells, right? So there are regular pili, pilus, P-I-L-U-S is the singular form, pili, P-I-L-I is the plural form. There are regular pili, and then there are special pili that only some bacteria produce that are particularly sticky, and we'll talk about those. So two types of adhesion factors are fimbria and pili that only certain types of bacteria produce, and even if they produce them, some of them are more sticky than others, and we'll talk about that. All right, we need to learn these. I will probably ask you multiple choice questions about these at some point in the future. All right, so S. pyogenes, Streptococcus pyogenes is a bacteria that we're going to hear about again and again, and we'll hear about it over and over again, so you may as well learn the name. So Streptococcus pyogenes, some strains of Streptococcus pyogenes produce a protein called protein F, which is an adhesion factor that allows it to stick to respiratory epithelial cells. Respiratory epithelial cells is a fancy word meaning lung cells and cells in the bronchial tubes, right? So some virulent strains of Streptococcus pyogenes produce a protein called protein F that allows them to stick to the cells inside your bronchial tubes or your lungs more easily, thus making certain strains of Streptococcus pyogenes easily transmitted by droplet transmission, for instance. Streptococcus mutans causes tooth decay, and one of the reasons it causes tooth decay is because it produces a virulence factor called adhesin P1. So that's a protein that allows it to stick to the enamel of teeth very well. Okay, Neisseria gonorrhea is a bacteria that causes a sexually transmitted disease called gonorrhea, and it produces one of those special types of pili that I mentioned in the previous slide. It produces something that's called a type 4 pilus, plural type 4 pili. Right, so a type 4 pilus allows Neisseria gonorrhea to stick to urethral epithelial cells. Right, the urethra is the tube that leads from the bladder out of the body. Right, so the urethra is in the vagina and in the penis, which the fact that the urethra is right there by the sex organs makes it ideal, makes Neisseria gonorrhea ideal as a sexually transmitted disease because it sticks to the urethra. It sticks to the cells of the urethra. And the reason it does that is because it produces a specialized type of pilus which can be used for mating with other bacteria, but it can also be used to attach itself to epithelial cells in the urethra. Okay, now, enterotoxic E. coli, abbreviated ETEC, produces a type of fimbria. Remember we talked about fimbria in the last slide. It produces a special type of fimbria called type 1 fimbriae, which specifically allow that type of bacteria to attach itself to the intestinal epithelial cells, which are found on the inside of the intestine. Right? So, all right, that's what it uses to adhere to the host cells. And then Vibrio cholera produces another special type of pilus, which is referred to a pili that's made of a special protein called N-methylphenylalanine, N-methylphenylalanine pilus, right? So these are examples of specific attachment or adhesion factors, rather, that I might ask you about on multiple choice questions. Okay, let's look at some examples of exoenzymes. Exoenzymes are catalysts that are secreted by certain bacteria that usually break down tissues and make it easier for the bacteria to spread throughout the body more freely. These are not adhesion factors. These are things that make invasion more easy, right? So adhesion factors are used to make adhesion more easy. Exoenzymes are used to make invasion more easy. And remember that we go from exposure, adhesion, invasion. So exoenzymes they come into play during the invasion phase of infection. All right, here's some examples of exoenzymes. Bacillus anthracis produces an exoenzyme called phospholipase C that breaks down the phospholipid bilayer and lyses cells or breaks open cells, causing bleeding, causing the formation of ulcers. So you have these bleeding ulcers that are kind of what we call weeping ulcers, because they also allow a lot of fluid to seep out of the ulcer. And that's a result of the Bacillus anthracis producing this enzyme called phospholipase C. Clostridium perfringens produces a number of different proteases that break down the tissue, the surrounding tissue, and causes a disease called gangrene. So when you get a wound that's infected with gangrene, with Clostridium perfringens, it turns black, and in the old days, they used to cut the limb off if you had a limb that turned black like that, because if you didn't cut it off, it would continue spreading to the rest of your body and kill you. So that the only way they knew of to stop it in the old days was to cut off the limb. These days, you can cure it with antibiotics sometimes, not always, but sometimes. Okay, then Staphylococcus aureus produces a bunch of enzymes that are referred to as glycohydrolases that break down a chemical called hyaluronic acid. If you took Biology 130, you know that hyaluronic acid is one of the main constituents of connective tissue. Right, so connective tissue, loose connective tissue, fibrous connective tissue, these connective tissues that hold your body together contain a lot of hyaluronic acid, and Staph aureus can break down the connective tissue in your body, allowing themselves to spread more easily. Now, not every strain of Staph aureus produces those, but if they do, they can cause a really nasty thing called flesh-eating bacteria. We label them as flesh-eating bacterial disease. That happens occasionally, and it's very dangerous. We don't know why some strains of Staph aureus are producing the glycohydrolases and most are not, but some do, and the ones that do are particularly dangerous. Okay, so breaking down the connective tissue allows the bacteria to spread throughout the body more easily. Okay, breaking down the connective tissue also allows the bacteria to get into the bloodstream and then travel around the body, and then you're really in trouble if the bacteria starts traveling around in the bloodstream. Okay, toxins. Let's look at exotoxins and endotoxins. Okay, an exotoxin is a toxic substance that's, we'll start with endotoxins. An endotoxin is a toxic substance that's an integral part of the bacterial plasma membrane. It's only dangerous if you kill the bacteria. So, for example, we talked about gram-negative bacteria. They produce a toxin, an endotoxin called lipopolysaccharide, or LPS. You do need to know that term. So you need to memorize LPS and know the fact that gram-negative bacteria have LPS in their plasma membrane. And if you kill them, if they infect your blood, if you kill them, they will dissolve and release that exotoxin and kill you. So here's what LPS looks like. LPS is actually composed of 3 different sections. One section is called the O antigen, which is highly allergenic, and so it causes toxic shock. It causes anaphylactic shock. And then the lipid A allows it to anchor into the plasma membrane of the bacterial cell, and then there's a part called the core. It's actually the O antigen that's the worst part of it, right? So LPS, if I ever asked you, give me the most toxic component, tell me which types of bacteria produce endotoxins, and give me an example of an endotoxin, you would say example lipopolysaccharide, example of a bacteria that makes it, Escherichia coli, or in fact any of the gram-negative bacteria, but Escherichia coli, E. coli is a good example. And then I ask you, what is the most toxic part of the LPS? You would say the O antigen. Okay, exotoxins, toxins that are sent out to kill the host. All right, there are several different types of exotoxins. The most popular type, if I can put it that way, the most popular type is called an AB exotoxin. And what happens with an AB exotoxin is that the bacteria, it's usually bacteria that make these type of toxins. The bacteria makes the toxin in two parts, two separate parts. It's a modular toxin, if you will. We call the different parts Part A and Part B. Part A is the toxic part, and Part B is the adopter, basically, that binds to a receptor on the surface of the cell, right? So Part B basically is targeting the toxin to the cell, right? So Part B is produced, it's secreted outside the cell, and Part B attaches to a cell surface, and then Part A, which is the toxic part, will attach to Part B and therefore kill the cell. Okay, so those are AB toxins. We talked about membrane disrupting toxins when we said, phospholipase C, for instance, destroys the phospholipid bilayer. That's an example of a membrane disrupting toxin. Not all of the exotoxins work that way, but many of them do. And then thirdly, some exotoxins are what are known as superantigens. Right? So a superantigen is something that causes anaphylaxis, causes anaphylactic shock, which we talked about a few minutes ago. So the LPS endotoxin is a good example of an exotoxin that works that way. Okay, so here we have the AB exotoxin, where the bacteria, usually it's a bacteria that produces the toxin in two separate components. One of them targets the surface of the cell, that's the B component usually, and then the A component attaches to the B component, and it's the A component that's actually toxic. I'm going to give you some specific examples of AB toxins, and you should memorize these because I'll probably ask you at some point about these on tests. Right? So Vibrio cholera causes a disease called cholera, of course, and it produces an AB toxin that's produced in two parts, and the toxic part causes a lot of a substance called cyclic adenosine monophosphate, or cyclic AMP, to be produced in intestinal cells. Now, one of the things that cyclic AMP does is it causes a lot of fluids to be secreted from those cells, and if you have a lot of fluids that are being secreted from intestinal cells, that causes diarrhea. So the main symptom of cholera is diarrhea, and people who, if you have diarrhea, you lose a lot of water, right? So you lose a lot of liquid. Humans cannot tolerate losing a lot of water. If we lose about 10% or 20% of our water budget, that's lethal. That would actually kill us. So if you have a lot of diarrhea, you lose a lot of water, and people die from cholera due to dehydration. It stands to reason, therefore, that you can prevent people from dying of cholera if you give them lots of water to drink. So they have diarrhea, they won't be very happy, but if you give them lots of water to drink, the water that they're losing through the diarrhea will be replaced. However, if the water that you're giving them to drink is cholera contaminated, that's just going to make the diarrhea worse. Right? So one of the most cheap and common ways and best ways to treat somebody that has cholera is to give them lots and lots of clean water to drink. And you get clean water either by bringing in bottled water or by filtering the water. So it is possible to buy filters that filter out the Vibrio cholera, so that's another thing you can do. All right, now we mentioned the fact that Clostridium tetani causes a disease called tetanus. The toxin, these bacteria produce a neurotoxin that causes rigid paralysis. right, that causes you to become stiff, causes all the muscles in your body to become stiff, including the diaphragm. So the diaphragm, the diaphragm is stiff and flexed, you can't exhale, right? So then you die of suffocation basically once in a while. But generally, if you get to the hospital, they put you on a ventilator, so the machine helps you to breathe. Okay, now how does this toxin, basically it's called tetanus toxin, as you would guess, called tetanus toxin, and it's abbreviated capital T, small e, capital N, capital T, TNT. Right? It inhibits the release of a neurotransmitter that tells the muscles to relax, right? So, okay, and then Clostridium botulinum produces botulinum toxin, which is commonly referred to as Botox, and it's abbreviated capital B, small o, capital N, capital T, commonly referred to as Botox, which causes flaccid paralysis. Flaccid paralysis means loose or limp, right? So, flaccid paralysis, the toxins, the two Clostridium toxins, tetanus and Botox, are examples of AB toxins. And there's a bacteria called Corynebacterium diphtheria, which causes a disease called diphtheria, as you would guess. And it also produces a toxin called diphtheria toxin, which is also an AB toxin, right? So we have 1, 2, 3, 4 different toxins produced by 1, 2, 3, 4 different microbes that are examples of AB toxins. Let's talk now about membrane disrupting toxins. We've already talked about some of them. Okay, so Strep pyogenes produces streptolysin, which is a membrane disrupting toxin. Streptococcus pneumonia produces a toxin called pneumolysin, which is again a membrane disrupting toxin. If you get streptococcal pneumonia, you develop a terrible cough, it infects your lungs, and you develop a terrible cough, and you start coughing up blood because the Streptococcus pneumonia that's in your lungs is breaking down the lung tissue cells, and the capillaries that surround your lungs are bleeding into your lungs, and so you end up coughing blood. Staph aureus, Staphylococcus aureus, produces something called alpha toxin. So does Clostridium perfringens. And Pseudomonas aeruginosa also produces phospholipase C, which we heard about before. Okay, an antigen, a superantigen, is something that causes an allergenic reaction, an allergy, which is also known as a hypertension reaction. If you have a violent allergic reaction, it's called anaphylaxis. Right, so you may know some people who are allergic to peanuts or bee stings. They have peanut allergies, and if you feed them a peanut, this happens. You can see that the eyelids are swollen shut because the eyelids are made of soft tissue, and so if the capillaries become leaky and allow the serum to leak out into the soft tissues, you get swelling. And if all the liquid, or if a large portion of the fluid, the serum in the plasma in your blood leaks out, you have a drop in blood pressure. A rapid drop in blood pressure can sometimes be fatal. And so a toxin that causes a sudden violent allergic reaction can sometimes be fatal, and we call that anaphylaxis. You may know some people, if they have a peanut allergy, they have to have one of these things. They carry around one of these things called an EpiPen. You should know how to use that if you ever have to. So if you have a friend who has allergies and they've told you about it, they tell you that they're carrying an EpiPen. If they accidentally eat a peanut or get stung by a bee, depending on what their allergy is, you may be the one who has to rummage through their backpack and find their EpiPen. The saying is blue to the sky, orange to the thigh. What you do is you take that EpiPen and you take the cap off, and then you put the orange part against their thigh and you jam it into their thigh, and there's a spring-loaded syringe that's filled with epinephrine that causes their heart to beat faster, which brings the blood pressure back up. And it's an emergency way of saving somebody's life if they're dying from anaphylaxis. Okay, so some people have allergies to peanuts or bee stings or things like that, but virtually everybody has a violent allergic reaction to lipopolysaccharide, the LPS, and the O antigen. So that's how septicemia and bacteremia are bad for you. So there's LPS, the classic endotoxin, which gram-negative bacteria have. Okay, so two examples of these are two examples of other superantigens, right? So these two bacteria, Staphylococcus aureus and Streptococcus pyogenes, do not have LPS. The reason I mention that is because I just said that LPS is produced by gram-negative bacteria, and you will need to know this later on for the course, and that is that both Staphylococcus aureus and Streptococcus pyogenes are both gram-positive bacteria. They don't produce LPS, but they produce two other endotoxins. Staphylococcus aureus occasionally produces, depending on which strain it is, it will produce a toxin, an endotoxin called toxic shock syndrome toxin, TSST, toxic shock syndrome toxin. This is an unfortunate, sad story in the history of human endeavors. In the 80s, a number of women died when they used a type of tampon called Rely tampons. And the epidemiologists went and said, what do all these women who died have in common? It turned out they were all using Rely tampons. It turned out that the particular batch of Rely tampons that they used was contaminated with Staphylococcus aureus that was producing toxic shock syndrome toxin. So that caused them all to die of anaphylactic shock, and so they took that product off the market. But it wasn't something that was specific to the tampons. It was something that's specific to Staphylococcus aureus. So if the tampons had not been contaminated with Staphylococcus aureus, that wouldn't have happened. Next, Staphylococcus pyogenes. It's a bacteria that we'll talk about in detail later because it causes a number of very bad diseases. So you should remember the name Staphylococcus pyogenes. And it produces an endotoxin called streptococcal mitogenic exotoxin. Okay, it is actually that's an exotoxin, not an endotoxin, but it is an example of a superantigen. Okay, so this slide shows you that so we now know that E. coli produces a superantigen which acts as an endotoxin, and that superantigen is called lipopolysaccharide. And two other examples of superantigens, one is produced by Staph aureus called toxic shock syndrome toxin, and the other is produced by Streptococcus pyogenes, which is called streptococcal mitogenic exotoxin. Those are two other superantigens that you should be familiar with. Okay, and Staphylococcus aureus and Staphylococcus pyogenes are both pyogenic bacteria that cause the production of pus. Okay, so I mentioned earlier that pus is the remnants of dead phagocytes that are there to eat bacteria, and we classify any bacteria that will automatically provoke the formation of pus. We classify them as pyogenic bacteria. All right, let's discuss how to avoid getting stuck or killed by the immune system. Right now, the term antigen you should be familiar with. The term antigen is a foreign protein that you find on a foreign body or foreign microbe that provokes the formation of antibodies. They are the cause of antibodies being produced, and therefore they are an antigen. Right? So you know that your immune system is able to recognize your proteins, and it is able to distinguish your proteins from foreign proteins. And so if a microbe gets into your body that's covered with foreign proteins, your immune system will get to work destroying it because it's able to recognize that those are foreign proteins, right? So those foreign proteins are referred to often as foreign antigens. Foreign antigens. Okay, now, how do you avoid the immune system if the immune system is able to spot foreign proteins? Well, there's a couple of ways. Okay, so remember that when the immune system attacks foreign invaders, it's recognizing the foreign proteins. The human immune system is very good at seeing and recognizing foreign proteins, but it's not particularly good at recognizing foreign carbohydrates. Okay, so one of the ways that a bacteria, for example, one of the ways that many forms of bacteria can avoid the immune system is by producing something called a glycocalyx. A glycocalyx is a layer of polysaccharides that the bacteria uses to surround itself and hide its proteins underneath it. It's kind of like putting on a cloak. It puts on a cloak of polysaccharides to hide the foreign proteins that it has on its plasma membrane surface. Now, there are two types of glycocalyces. One is called a capsule. A capsule is if the carbohydrates, the polysaccharides that make up the glycocalyx, if they're well organized, they're kind of hard and inflexible, and we call that a capsule. If the carbohydrates that are making up the glycocalyx are loosely organized and disorganized, they're sticky but not hard, we refer to that as a slime layer, right? So glycocalyx is an umbrella term that refers to, collective term that refers to both a capsule and a slime layer. So a capsule and a slime layer are both examples of a glycocalyx. And a glycocalyx is something that bacteria use both as an adhesion factor and also as a way of evading the immune system. So I should mention also that a slime layer and to a certain extent a capsule are also adhesion factors because they help the bacteria to stick to the cells. The slime layer in particular is particularly sticky, and so a slime layer is not only a way of avoiding the immune system, but it's also a way of sticking to the host. All right, now, antigenic drift. This is something that is unique to RNA viruses. I mentioned the fact that many viruses have a genome that's made of DNA, but some viruses have an RNA genome. For example, the coronavirus, the COVID-19 virus has an RNA genome. Now, one of the things about RNA genomes is that when a virus enters a cell, if a virus has a DNA genome, human cells, for example, and the cells of other animals have enzymes in their nucleus that are referred to as proofreading enzymes. Proofreading enzymes, and so if any of those genes, DNA genes, become mutated, these enzymes help to repair some of the mutations. As a result of that, organisms that use a DNA genome, their proteins do not change very quickly. Right? The proteins in an organism with a DNA genome do not change very often because any mutations that would cause changes to the structures of those proteins are fixed. Any mutations that would cause changes to those proteins are fixed by the DNA repair enzymes. Okay, now, if you have an RNA genome, there are no enzymes that repair mutations to RNA. So RNA genomes will sustain mutations more often, and that will lead to changes in the structures of the proteins. Okay, the problem with that is that you get infected with an RNA virus, your immune system goes to work at killing it, but if during that process the virus changes due to a mutation to one of its genes, the proteins on the surface of that virus may change enough that your immune system is not ready for it, and it has to start again by going and getting more B cells that can attack that particular surface protein. Right? So when your immune system attacks a foreign protein with antibodies, that process takes about 2 weeks. Right? So you may remember from first year that what happens is you have a foreign bacteria that comes in, it's covered with foreign proteins, it gets eaten by a phagocyte. The phagocyte is then renamed an antigen-presenting cell, an APC. A T helper cell will then come and see the antigen that is being presented by the APC and then go and get some B cells that are capable of making antibodies that are capable of binding to the foreign proteins that are on the surface of that bacteria. That's a very complicated process that takes about 2 weeks to carry out. Okay, so supposing a virus gets into your body, that virus will be eaten by an antigen-presenting cell, and the T helper cell will go and get some B cells, and then it'll make some antibodies that can destroy that virus, and that takes about 2 weeks. Fair enough. Fair enough. That takes about 2 weeks. What happens if during that 2 weeks the virus actually changes the foreign proteins because it sustained a mutation or two? Then your immune system has to go back and start again, and it has another one of these new variations of the virus has to be eaten by an antigen presenting cell, and then which is then transmits the information to a T helper cell and gets some more B cells. So by changing the proteins on your surface, that's a way of staying ahead of the immune system. And so if it takes the immune system 2 weeks to catch up to you, if you have some way to change the proteins on your surface, ideally during a time period of less than 2 weeks, then you can stay ahead of the immune system. Okay, so now as it happens, RNA genome viruses don't change their proteins that fast. They do change their surface proteins once in a while, and that's why we have to keep on, we have to keep on getting a new vaccine every year for the flu, because the proteins that are on the surface have been changed on average about once a year. We call that the seasonal flu. All right, now there's another way that RNA viruses can change their genomes very fast, and that's referred to as antigenic shift. All right, now antigenic shift is where you have an RNA virus that has an RNA genome, but the complete genome is stored on more than one chromosome. We call it a segmented genome. Right, so you know from first year biology that the reason we look different than our parents is because when our parents created us, they donated gametes that had the alleles for all of our different genes shuffled and rearranged, right? So it's like shuffling a deck of cards. You get a different combination of gene alleles every time. That's only possible because the chromosomes sort independently, right? So independent assortment of chromosomes is one of the main reasons why each of us looks different than our parents. And we have many proteins in our bodies that are different than the proteins that either of our parents had. It's because of independent assortment of the chromosomes, right? So some viruses can do that too in a slightly different way. So there are many viruses that just have, in fact, most viruses just have one chromosome. But there are some viruses that have more than one, and we call that a segmented genome, which means that the viral genome is on several chromosomes. For example, the influenza virus has 8 chromosomes. It has an RNA genome, and that genome is distributed amongst 8 different chromosomes. Now, because it's an RNA virus and it's subject to antigenic drift, that means that you can have 2 different strains of the influenza virus which have 2 different, slightly different sets of proteins, and those proteins are encoded by genes that are on 8 different chromosomes. If a person or an animal is co-infected, is simultaneously infected by 2 different strains of the influenza virus, that means that those chromosomes can be rearranged and come back together in a different combination. They can be reassorted. They can sort independently, and then you end up having in a very short period of time, you've created a totally new influenza virus that has a totally different combination of proteins on its surface, which your immune system is not ready for. And in fact, that's what happens with the flu virus every now and then. We have a big pandemic from the influenza virus because a person or an animal has been co-infected, simultaneously co-infected by 2 different strains of the influenza virus, and the chromosomes have been reorganized, reassorted to create a totally new version of the influenza virus. All right, so that is the definition of antigenic shift. Okay, what about antigenic class shifting? All right, there are some bacteria and some protozoa that have genes that encode several proteins. A good example is the protist called trypanosomes, the genus Trypanosoma. Right, so we talked about trypanosomes causing sleeping sickness, a disease called sleeping sickness, and we're going to talk about that more later. But for now, you should know that trypanosomes have about 100 different copies of genes that encode different proteins on their surfaces. And about every 2 weeks, it takes about a week or so for trypanosomes to give birth to a new generation, which means that if you're infected with trypanosomes, they have one set of proteins on their surface. Those trypanosomes will reproduce every week or so, and the next generation will have a totally different set of proteins on their surface because the trypanosomes are turning some genes on and other genes off. There's a whole lot of scientific research going on at the moment to understand how and why trypanosomes are able to turn different subsets of genes on and off in order to vary which proteins are displayed on their surface. We don't know exactly how they do that. It must be a very complicated process. We don't know exactly how they do that, but the reason it's important is because that's a way that trypanosomes can stay one step ahead of the human immune system. It's because the human immune system comes and sees what the foreign proteins look like on the surface of those trypanosomes, and then the T helper cells go away and find some B cells that can make antibodies that will stick to those proteins and help to destroy those trypanosomes. And after they've gone through all the trouble of doing that, the trypanosomes have changed the proteins on their surface. And so then the immune system goes back and gets more B cells to react to the new proteins, and then they've changed again. And so the trypanosomes, by turning these different subsets of surface protein genes on and off, they are able to stay one step ahead of the immune system. That's called antigenic class switching. All right, let's look at some specific examples of these different techniques and the different microorganisms that practice these techniques. Okay, so as I said, a glycocalyx is a polysaccharide shell, a polysaccharide cloak that is used to hide the viral or the bacterial, usually bacterial proteins. So Streptococcus pneumoniae, we've just discussed that a little bit, it produces a capsule. Some virulent strains of Streptococcus pneumoniae produce a capsule which helps it to stick to the lung cells as well as evading the human immune system. Streptococcus mutans, which causes tooth decay, produces a slime layer which also helps it to stick to the tooth. Right, so those are two specific examples of bacteria that make glycocalyces. Right, so here you can see the bacterial cell inside this nice slime layer or capsule of polysaccharides, and so all of the proteins on the surface of that bacteria are hidden underneath this glycocalyx, making it difficult for antibodies or for T helper cells or for phagocytes to spot them. Okay, RNA mutates faster than DNA because we don't have any proofreading enzymes to correct mutations. The result of this is that the proteins on the surfaces of viruses that have RNA genomes change fast enough to keep ahead of the immune system on a global scale, right? So a good example of this is the rhinovirus, which causes colds, right? So colds are caused by a type of virus called a rhinovirus. Rhinovirus has a single chromosome, right? So the rhinovirus does not do antigenic shift, but because it's an RNA virus, it does do antigenic drift. Drift, the word drift implies kind of a slow movement, right? So if you have a ship that's drifting, it means it's moving slowly in a kind of a random direction. So antigenic drift means that the genes of an RNA virus that has a single chromosome undergo these mutations every now and then. That causes the proteins on their surface to change, and you've created what's known as a new serotype. You've created a new serotype of the virus. There are actually 100 different serotypes of rhinovirus, and you never catch the same serotype twice because you've become immune to it. But nevertheless, you're catching a cold all the time because there's so darn many of them. There are 100 different serotypes of rhinoviruses. You catch one serotype, you get over it, you're immune to it, then you catch another serotype a few months later, and then you go through the whole process again. That's a result of the fact that rhinoviruses, there are 100 different serotypes of rhinoviruses. The reason there are so many serotypes of rhinoviruses is because the RNA genome has been mutating over millions of years, and we've ended up with 100 different variations of it. Now, this diagram is actually showing influenza virus that has 8 chromosomes, and because influenza has an RNA genome as well, you would expect some of the gene alleles to be different, right? So if you look at the top chromosome, we have 2 different alleles of the same gene. Now, if somebody gets co-infected with simultaneously co-infected with 2 different strains, this chromosome could end up being paired up with these chromosomes, and that would be a rearrangement of the chromosomes, which would lead to the creation of a new strain of influenza. By the way, if you look at this diagram, these proteins that are on the surface, these things that are called viral spikes or viral attachment proteins that are on the surface of the influenza virus, they allow the virus to attach to the cells inside your lungs. Therefore, those are actually adhesion factors. Those are virulence factors. And there are 2 different types of viral attachment proteins that you find on the surface of an influenza virus. One of them is called hemagglutinin, beginning with the letter H, and one is called neuraminidase, beginning with the letter N. Do the letters H and N sound familiar to you in the context of influenza virus? In the context, you've heard of H1N1, for instance, H2N2, H1N5. It just so happens that the influenza virus has multiple alleles of the gene encoding the hemagglutinin protein and multiple alleles of the neuraminidase protein that are present in different strains of the virus. And we label them N1 through N8 and H1 through H13. And then if you get a co-infection, you can get a rearrangement. So you co-infect cells with H2N2 and H1N1, and then you may come out with H1N2, right? Because the chromosomes have been rearranged. So that's exactly what we're talking about when we talk about the H1N1 Spanish flu epidemic, for instance, pandemic. That's exactly what the H1 and the N1 are referring to. They're referring to alleles of the hemagglutinin and the neuraminidase genes. All right, antigenic shift is what I just explained. Right, so you have viruses that have RNA genomes. Those genomes will change over time, giving you different alleles, for example, the hemagglutinin and the neuraminidase. And then those different alleles, the genes, the different alleles are carried on different chromosomes. If somebody is co-infected with two different strains of the same virus that have different alleles, you could end up having chromosome reassortment, which leads to a totally different combination of alleles, which leads to a totally different flu virus. All right, just like you see here. So here you have, here we have two different strains of the influenza virus. Somebody's co-infected with the two different strains, and then we get a different arrangement of the chromosomes leading to a totally different virus. And that's actually what happens when we have these pandemics, influenza pandemics, like the 1918 we had the Spanish flu pandemic. We've had the H1N1 flu pandemic a few times since, and I venture to say that we are going to have H1N1 and various other influenza epidemics in years to come. We need to be prepared for these kind of epidemics because they're going to happen all the time, because the conditions are not changing. The conditions for creating new strains of the flu virus are not going away. So we will in the future have more pandemics of the influenza virus. So governments need to be prepared for this. If you ever go into government and you are in a position to make these kind of decisions, you should be aware that we are going to suffer from influenza pandemics with some frequency in the future. And so you should think about making government policy to accommodate this. All right, antigenic class switching. Trypanosomes are a protozoa. Trypanosoma brucei you would find in Central Africa. Trypanosoma cruzi you will find in Central America. These trypanosomes have several different genes that code for proteins on the surface, but they don't have them all turned on. They have some of them turned on and some of them turned off, and they change which ones are turned on versus turned off every generation in order to keep ahead of the human immune system. So this is what they look like. Over here we have Trypanosoma brucei, which you find in Central Africa. It causes a disease called sleeping sickness, and it is spread by a vector. It's spread by an insect vector called the tsetse fly. There it is. And then over here we have Trypanosoma cruzi, which is found in Central America, and it is spread by an insect vector called a kissing bug. Right, and in either case, these are two very similar strains. Now notice this beautiful picture of the trypanosome here. Notice that you see the nucleus in the middle. That reminds us that this is a single cell, right? This is just one cell. It's not a multicellular organism, but look at how complicated it is on the right-hand side. You see this, it has a kind of a fin to swim around, right? And it's a single cell, but it's very complicated. It has this kind of a curly fin on the top called an endoflagellum, which allows it to swim around. And all right, so these interesting organisms can practice antigenic class switching, where every new generation has a different combination of proteins on its surface, which allows it to stay ahead of the immune system. So you get infected with what's known as clone A. A clone is like, a bunch of them that are all the same. So you get infected with one of them that's a member of clone A. Your immune system takes 2 weeks to make antibodies that'll destroy it. And when those antibodies are ready, the stupid thing is changed to another set of proteins. Your immune system says, darn it, I need to start again. It goes and makes new antibodies to destroy this one. And when it comes back, suddenly that one is there and this one is all gone. And this recurring the recurring change of this immune reaction is what causes recurrent fevers. So every 2 weeks you have a new generation of trypanosomes being born, which leads to a new fresh immune reaction which causes a fever. Okay, now as promised, let's talk about the periods of disease. Right? So periods of disease refer to different stages of the illness. We've talked about the different stages of the infection. We talked about exposure, adhesion, invasion, and so on. Now let's talk about what happens when you actually get sick. All right, typically, once you've been invaded, once you've been infected, you've got a full-blown infection, your illness will take the following course. The first period of illness is referred to as the incubation period, and this is a critical period because you have significant numbers of microbes, either viruses or bacteria or fungi or whatever. Let's talk about viruses because they tend to be contagious. You have an incubation period where you have significant numbers of microbes in your system, in your blood or your other bodily fluids, but you are asymptomatic. You don't feel any symptoms. If it's a contagious disease like a virus, a droplet transmission virus, if it's a contagious disease, you will be contagious during the incubation period even though you don't realize you're sick. All right, the prodromal period, the second phase, is when you still have significant numbers of viruses, for example, in your blood, and you're just starting to feel sick. So when you have a cold, that's when you're starting to have a scratchy throat and a cough. Then, at that point, you know that you're sick, and usually you'll either put on a mask to avoid making other people sick, or you'll stay home. Okay, then you get into the peak of your symptoms. That's called the period of illness. You have the maximum amount of symptoms, and you are also most contagious. Then, after you get over that, your immune system has started to destroy the invader, and then you get a period of decline, so the symptoms are starting to feel better. And then when you're starting to feel almost back to normal, you have what's referred to as the period of convalescence. The length of time that you spend in each of these periods varies depending on which microbe we're talking about. Right? So bear in mind that you never spend, I'm not showing you any time periods here because the time period that you spend in each of these phases depends on which illness we're talking about. Now I want to show you this wonderful graph here. We have a red line and a blue line. The red line is showing you the number of pathogens in your system, the number of virus particles, for instance. The blue line is showing you the severity of your symptoms. And up here we have the period of incubation, the incubation period. And I want to show you this down here, that you have significant numbers of viruses in your blood or bacteria or whichever illness we're talking about, but you don't have any symptoms yet. Right? So that's the period of incubation, and that's a particularly important period for contagious diseases because you are contagious, but you don't know you're sick yet. Right? So it stands to reason that if you have a long incubation period for a contagious disease, that disease will be particularly menacing because you're going around spreading it to people for a longer period of time before you realize that you're sick and therefore take measures to prevent spreading it to other people. Right? If a contagious disease has a short incubation period, that's a good thing because you're not spreading it to other people for a long period of time before realizing that you're sick. Right? So the incubation period is critical, and that's something that you need to memorize, and because it's important. I'd like to draw your attention to one other thing here. There's a myth that you might have heard where somebody shows up at work and they're coughing and they're sneezing and they say, "Don't worry, I'm not contagious anymore." There's a myth that during the maximum period of symptoms, you are not contagious anymore. But this graph proves that that's not true. Look at the red line. The number of particles, the number of infectious particles that are in your body are greatest when you have the greatest symptoms, right? So when you feel the sickest, that is the period when you're the most contagious, right? So it's exactly the opposite of what that myth says. You know, we call them urban myths or urban legends. This urban myth says that when you're in the deepest part of a cold, when your nose is running and you're coughing and sneezing, you're not contagious anymore. That's actually a myth. It's not true. You're actually the most contagious at that point. All right, so that's that. Let's look at the incubation period some more. At the incubation period, you're contagious, but you have no symptoms. This is a dangerous period because you're spreading the disease without realizing it. Okay, during the prodromal period, you're starting to feel the symptoms, and you will probably stay home or wear a mask to work if we're talking about a respiratory virus like COVID-19 or influenza or a common cold. All right, now here's something interesting. With the COVID-19 crisis, here's something interesting. They introduced these measures. One of the public health measures that they've introduced is to start measuring people to see if they have a fever, because one of the symptoms of COVID-19 coronavirus is that you have a fever, you have an elevated body temperature. Now, one of the ways that you can stop people from spreading the virus is to check and see if they have it. And if they have it, you put them in quarantine, or at the very least, you don't let them get onto a crowded airplane with a bunch of other people. So at the beginning of the COVID-19 crisis, many countries started doing this. They prevented people from going on airplanes if they had a fever. Okay, that's fine. So you don't want to put a thermometer in everybody's mouth and measure their temperature, because then the thermometer would become a fomite, wouldn't it? So that the thermometer would actually be spreading the disease rather than just measuring it. So what they did was, they have this thing here is an infrared camera that can measure the temperature of your forehead without actually touching your forehead. So you might have seen these at airports where you have safety people are checking everyone's temperature before they go on the plane, and if you have a fever, they say you can't go on the plane, you have to go into quarantine. Okay, that's fine, but generally with COVID-19 and with many viruses and things like that, you don't get a fever until you're already in the prodromal period, right? So what about all these people that do not have any symptoms, they don't have a fever, and you just let all of those people onto the plane, didn't you? Right, so checking for the fever is useful to a point because you're catching people that definitely have the disease, but you are failing to catch a bunch of people who have a disease and who are contagious but who happen to be in the incubation period. So many countries were doing this. I say that it's not completely useless because it's a way of catching people who are sick, and then you put them into quarantine. And if you have a contagious disease and there's no other way to contain it, the only thing you can do is there's no other way to defeat it. The only thing you can do is work on measures, large-scale public measures that are meant to contain the contamination. So you break the chain of contamination by isolating people who are sick and keeping them away from people who are not sick. And one of the ways that you can do that is you can set up random roadblocks and you can measure people's temperature. You can set up you can set up taking everyone's temperature at the airport. And that is a way of determining that's a very crude and somewhat ineffective way of determining who's sick, so that gives you the option of taking them out of the population and quarantining them to try and break the chain of contamination. Right? It's not the best way because it doesn't account for the incubation period. All right, you might have heard a lot of talk in the media about PCR testing for coronavirus. PCR stands for the polymerase chain reaction. Some of you know what that is, some of you don't. For those who don't, just suffice it to say that a PCR reaction is a way of checking for the presence of genes from a virus, right? It's a way of, you take a blood sample, or more commonly you shove a swab up somebody's nose and you get a sample of their mucus, and then you put it into a machine called a PCR block or a thermal cycler, and it will do some reactions, and then it will tell you if there are any genes from the virus in question present. Now, the nice thing about PCR tests, we talked about this when we were talking about Koch's postulates. The nice thing is that they are extremely sensitive. So a PCR test can detect very, very, very small numbers of viruses or bacteria. PCR test is extremely sensitive. Checking the fever is an extremely blunt, insensitive method, right? So a PCR test is very sensitive. Unfortunately, it's also very expensive, right? So that's why it's a trade-off. Checking people's temperature is very cheap, but you lose a lot of people. A lot of people fall through the cracks, as they say. A lot of people that are sick and who are contagious are not screened out because checking the temperature is not that sensitive. But checking for the presence of genes using the polymerase chain reaction is very sensitive, but unfortunately it's expensive. So when you're making public policy, you have to trade those two things off. Temperature testing and other methods are very cheap, but they miss a lot of people. So what if we just do something, if you're a politician or a public health official in a country that doesn't have a lot of money to spend, you can say, well, we'll test everybody using the cheap method, temperature, and we'll test only the important people using the PCR method. Example, you check the healthcare providers, you check the doctors and the nurses who are taking care of all the COVID patients to make sure that they haven't got it, because it makes more sense to check them because they are exposed to the COVID-19 virus more often than everyone else. And so they could become spreaders. They could become vectors, as it were, to spread the virus to other people unless we check them regularly using a very sensitive method to make sure that they don't have the disease. All right, you've heard of a quarantine period. The word quarantine comes from the Latin word quarantina, which means 40 days. That was a thing that the Italians developed during the period of the bubonic plague, where places like Venice in Italy, they would have foreign ships coming from all over the world occasionally. Those foreign ships would bring foreign diseases to Venice, and so they were required to hoist a plague flag to say that they had plague. They either had leprosy or bubonic plague, and that ship was kept in quarantine for 40 days for a quarantina. So they would throw food onto the ship for the sailors to eat, and then if nobody else got sick after 40 days, they could come ashore. You would assume they didn't have the disease if they didn't show any symptoms after 40 days. Now, the quarantine period these days will depend on the disease, and the quarantine period is designed to match the known incubation period for that disease. Okay, so if you have a disease that has a short incubation period, somebody's been exposed to it, you have no way to test whether they have it or not, but you know they've been exposed to it. In order to find out whether they've got the disease or not, you put them in quarantine, and if they don't show any symptoms after a number of days, then you know they don't have it, you can let them go. The incubation period, the number of days that you have to wait to see if they display any symptoms, depends on the disease. Right? So the quarantine period for diseases is meant to match the incubation period. So as I said, here we have ancient Venice in Italy. You have foreign ships coming in. If they had a disease on board, they had to run up this flag, which is called the plague flag, the quarantine flag, which means that there is a disease aboard that ship. Maybe a sailor got sick with some disease and they died and they threw him overboard, they buried him at sea, and then they want to find out whether the other sailors have the same disease. They have to quarantine that ship for 40 days. So that's the way they used to do it. You may know that during the early period of the COVID-19, many people on board these cruise ships got sick because cruise ships are floating incubators. It's very easy for diseases to be transmitted from person to person in a cruise ship. And guess what? It's also very easy for diseases to be transmitted from person to person in a hospital. And we referred to that at the beginning of this lecture as what? What did we call that? We call those nosocomial infections. So hospitals are great places to spread diseases. So hospitals have to take special precautions to try and limit the spread of these diseases, to try and limit the spread of nosocomial infections. All right, these are some of the incubation periods of common viruses. So notice that the influenza has an incubation period of about 2 days. So that's good, because if somebody gets the flu, they know about it after 2 days, and then they stay home and they stop spreading it to other people. COVID-19 has an incubation period of between 2 days and 2 weeks, depending. So the incubation period, when they were talking about these cruise ships not letting people off of these cruise ships when they knew that some people on board had COVID-19, they kept everyone on board the cruise ship for 14 days. And then if nobody else came down with symptoms of COVID-19, they would let everybody go. But if one person showed symptoms on day 13, they had to start the clock all over again and wait another 14 days. And so that's because the incubation period is meant to match the quarantine period is meant to match the incubation period, right? So COVID-19 is kind of bad because it has a relatively long incubation period. Not as bad as measles. So look at measles here. Measles has up to a 12-day incubation period, and it also has an R0 value of 18. So it's very contagious, and it also has a relatively long incubation period, which is why measles was particularly bad. Notice the mortality rate. The mortality rate refers to the number of people who are infected who will actually die from it. COVID-19 is very bad because it has about, according to the current data, as far as we know, it has a mortality rate of something like 7%, right? And other viral diseases like the seasonal flu do not, right? So the seasonal flu has a mortality rate of between 0.1 and 2%, depending on which strain it is. The mumps has a very low mortality rate, but it does affect children differently. Measles, there's an asterisk here. Measles has a 10% mortality rate if untreated. So if a child catches measles, there are various drugs and techniques that you can use to take care of them so that they won't die. But in the old days, children used to die of the measles. Okay, so this is just showing that any of these diseases, these different diseases have different incubation periods. If we have a PCR test to tell if somebody is sick with the disease or not, you don't have to quarantine them for that period of time. You can test them, and if they test negative for the disease, you can let them go. If you don't have a test, if you don't have a reliable test or a good test, you have to quarantine them for the period of time that is equal to the incubation period, which is why now you're hearing in the social media, for instance, if you've been traveling abroad and you come back to Canada, you have to self-isolate for 2 weeks, because 2 weeks is the period that you need to tell whether you've actually got COVID-19 or not. And then if you don't show any signs of COVID-19 after 2 weeks, then you can come out and you're not in danger of spreading it to other people. Okay, so that's the incubation period. That's why the incubation period is important. Okay, let's summarize what we know about microbial pathology, and then we'll have another lecture on epidemiology in which we will use some of these terms again. So we learned all about the resident microbiota of the human body and how generally those microbes are harmless, but many of them can cause opportunistic infections if for some reason your body is weakened, or if for some reason those microbiota are able to get into some place that you don't normally find them. Example, Staph aureus on the skin is harmless. If it gets into the vagina, it causes vulvovaginal candidiasis, Candida albicans. Gets into the vagina, it's harmless outside on the skin. If it gets into the vagina and there's no Lactobacillus acidophilus there to outcompete it, it will cause a yeast infection. Other example, if Staphylococcus aureus gets in through the parenteral route, through a break in the skin, it can cause an infection, formation of pus, and so on. We learned about the difference between signs, symptoms, and a syndrome. We learned what an asymptomatic infection is. Same thing as a subclinical infection. The two words mean the same thing. We learned what the word etiology and idiopathic mean. We learned the difference between an acute infection, a chronic infection, a latent infection, and a persistent infection. We learned the difference between local versus systemic infections. We learned about bacteremia and septicemia. We learned Koch's postulates. We learned a method of measuring the virulence of a particular strain of a microorganism by measuring the ID50 or the LD50. We learned about the stages of infection or the stages of pathogenesis, beginning with exposure, adhesion, invasion, and so on. If you're not exposed to tuberculosis, then adhesion can never happen because you weren't even exposed to it. But if you were exposed to it, it may not adhere. It may not stick to you. If it does stick to you, it may not invade. Some of the things that allow it to invade include the exoenzymes. Once it's invaded, some of the things that make it particularly nasty may be exotoxins or other exoenzymes. So all of those things we discussed. We discussed the various portals of entry. We discussed infection vehicle transmission versus vector transmission. We discussed vertical versus horizontal transmission, torch and transplacental infections, and teratogens. We learned about several specific examples of virulence factors which might be on some of the tests. If I ask you about those things, they will be multiple choice questions. You don't have to memorize them all completely, but they will be on tests. And we talked about the periods of illness. The most important period of illness is the incubation period, because that will generally determine how contagious a disease is, because it determines how long you're spreading it to people without knowing that they're sick. All right, the important concepts we learned about the World Health Organization. We learned the difference between communicable versus contagious diseases. We learned about the mortality rate versus the morbidity rate and how virulent things are. We learned about nosocomial and iatrogenic infections. We learned about zoonoses. We learned about the different modes of transmission, vehicles, vectors, the difference between a biological and a mechanical vector. We learned about vector transmission and a primary versus an intermediate host. Example, malaria, the actual primary host is the mosquito. The humans and the monkeys are only the intermediate host. Vector transmission versus mechanical transmission. So biological vector versus a mechanical vector means a biological vector is an insect or an arachnid that is actually infected by the disease that it's transmitting, whereas a mechanical vector is just a fly tracking things on its feet between piles of excrement and your food. Right? So we learned about a number of these concepts and a number of these terms. Now go on to the next lecture, and we will use these terms when we discuss the study of epidemiology.